Hello there. Welcome to Relax with Science, where the intricate poetry of the universe lulls you to sleep. You have had such a long day, and I know you must be so very tired. You have done enough. It is time, now, to simply rest. Let the heavy weight of the hours slip away, softening into the stillness of your room. Notice any tension you might still be holding, perhaps in your shoulders or around your eyes. Gently, let your jaw soften. Allow your limbs to grow heavy, sinking deeper and deeper into the soft bed beneath you. You are safe here. There is nothing left to do. Tonight, we are going to journey inward into a gentle, fascinating exploration of your own body. As we wander through this miraculous landscape, please remember, you do not need to memorize a single fact. You only need to listen and let the quiet perfection of the science wash over you. Something moved you to this quiet place tonight. Not metaphorically, but literally. The muscles of your legs, your hips, your core. The muscles of your arms. If you carried anything today, the muscles of your neck, which have gently balanced your head through all the long geography of the day. All of them brought you here. Every transition you made today, from lying to sitting, from sitting to standing, from one room to another, from the car, to the door, to the chair, to the bed, every single one of those movements was a muscular event. Every word you spoke, every breath you drew, even the soft blinking of your eyes across the hours of the day, muscular. And now, finally, having done all of this, they are beginning to let go. The great, voluntary unwinding of the day's last tension. The muscles releasing their holding, giving the body back to gravity, back to the soft surface that supports you, back to the particular stillness of the night. There are more than 600 muscles in the human body. 600 individual notes in a symphony of contraction and force, ranging from the massive, grounding muscles of the hip to the stapedius, the smallest muscle in the body, roughly 1 millimeter long, sheltered quietly inside your ear. There are the voluntary muscles you consciously command, and the smooth, involuntary muscle that lines your vessels and digestive tract, continuing its vital work without any instruction from you at all. Tonight, these muscles are our focus, though subject is perhaps not quite the right word. Companion is closer. Every fact we will visit tonight is a fact about something that is happening inside you, right now. The physiology, not of some theoretical body, but of your body. In this bed, in this dark, breathing this particular air, I find there is something quietly extraordinary about learning of the muscles at night, when they are beautifully at rest. When your body is horizontal, and the voluntary muscles of posture are finally released from their duty, and the heart muscle, ceaseless and so loyal, is pumping at its slowest, most peaceful rate. There is a gentle quality of recognition in hearing the facts of muscular physiology described in the dark that does not come in the bright, busy hours of the day. It is the difference between reading about the ocean while standing at the sea and reading about it while sitting comfortably, miles inland. Except that tonight, the ocean is inside you. We are going to travel somewhere remarkable together, slowly, gently, with nowhere else to be. Into the chemistry of contraction, and the delicate architecture of the muscle fiber, and the miraculous coordination of movement that the body performs so fluently, you have almost certainly never thought about it at all. Let the body be precisely where it is. Let your breath slow. And let us begin at the beginning, which is also, always, exactly where you already are. I wonder where you are lying down tonight. Whether the muscles of your back have found the particular ease that comes after a long day of holding. Whether your shoulders, so often the last to let go, have finally released a little of what they were carrying. Whether the quiet of where you are is absolute, or whether there is still a soft, distant sound in the world around you. If you feel like it, and only if you feel like it, tell me softly in the comments where in the world you are drifting off to sleep. A city, a country, a time zone, the temperature of the air outside your window. I read every single one. It matters to me that you are there. If these words bring you any ease tonight, any softening of the day's remaining grip, leaving a quiet like on the video is a gentle and generous way to say so. It helps our safe space find its way to someone else, who might be lying somewhere in the dark, needing exactly this kind of company. If you know someone who carries physical tiredness alongside all the other kinds, and most of us do, consider sending this to them. No explanation needed. Just the link, left where they will find it. And if there is ever something that could be softer, slower, or more beautiful about what we make here, I want to hear it. This channel belongs to the person lying in the dark. It has always been, for you. The human body contains more than 600 named skeletal muscles, and if you were to include the smooth muscle of the blood vessels and viscera and the cardiac muscle of the heart, the total number of individual muscle structures in the body climbs into the billions. But even the 600 named skeletal muscles represent a collection of force-generating organs whose combined capabilities define almost every action that a person can perform in the physical world. The number 600 carries a kind of weight that most people have not had occasion to feel. It is larger than most people imagine. The popular idea of the body's major muscles runs to perhaps 20 or 30 that most people could name. The biceps and the quadriceps and the abdominals and perhaps the gluteus maximus if they have had occasion to learn it. But the named muscles of the body are many hundreds more than this, ranging from the enormous to the minuscule. From muscles that span the entire length of a limb to muscles so small that their existence was not confirmed until the age of surgical microscopes. The sartorius, the longest muscle in the body, runs diagonally from the anterior superior iliac spine of the pelvis all the way to the medial surface of the tibia below the knee, spanning both the hip and the knee joints in its long oblique course. The stapedius, the shortest and smallest, is approximately 1 millimeter long and lives entirely within the ear canal, attached to the stapes bone of the middle ear, contracting reflexively to dampen the vibration of the ossicular chain when the ear detects very loud sounds, protecting the hair cells of the cochlea from damage. Between these two extremes lies the full range of human muscular architecture: the layered sheets of the abdominal wall, the paired rotator cuff muscles of the shoulder, the deep intrinsic muscles of the spine that make the fine adjustments to posture that no one consciously controls, the muscles of the tongue and the throat and the floor of the mouth that coordinate swallowing, the muscles of the eye that move the gaze with a precision measured in fractions of a degree. All of this is happening simultaneously, right now, at whatever level of activity the current moment demands. Most of it is happening without your awareness. Most of it has been happening without your awareness every moment since you were born. The 600 muscles are the machinery of the life you lead, operating so fluently and so continuously that their existence is only noticed when they ache, or when they fail, or when, as tonight, someone takes the time to describe them with the care they have always deserved. Muscle tissue is unique among the tissues of the body in its ability to convert chemical energy directly into mechanical work, a process that occurs at the level of individual protein molecules and that represents one of the most efficient energy-to-motion conversions in the known natural world. Every other form of mechanical energy the human body experiences comes from outside it: gravity pulling, wind pushing, the resistance of surfaces and objects. The only internal source of mechanical work, the only way the body generates force from within, is the contraction of muscle. And this contraction happens not at the level of the whole muscle, or the individual muscle fiber, or even the individual myofibril, but at the level of a single protein interaction, the binding of a myosin head to an actin filament and the subsequent power stroke that generates the force. This interaction between two molecules, each only nanometers in size, is the fundamental unit of all muscular force in the body. Every movement you have ever made, from the largest athletic effort to the smallest flicker of an eyelid, is ultimately the sum of millions or billions of these individual molecular events, occurring in coordinated waves across the muscle fibers, their collective effect appearing at the macroscopic level as the smooth, graded contraction of the whole muscle. The energy that drives this molecular interaction comes from ATP, adenosine triphosphate, the universal cellular energy currency. The hydrolysis of one molecule of ATP provides the energy for one power stroke cycle of one myosin head. The rate at which a muscle can generate force is ultimately limited by the rate at which it can produce ATP, and the various metabolic pathways by which muscles generate ATP. The immediate phosphagen system, the glycolytic pathway, the oxidative system, represent the muscle's different solutions to the problem of energy supply across different time scales and exercise intensities. What happens at the molecular level is, in a precise sense, Transformation. The chemical energy of ATP becoming the mechanical energy of movement. Efficiency in this conversion varies with temperature, with fiber type, with the specific conditions of each contraction. But at its best, skeletal muscle converts chemical energy to mechanical work at an efficiency of approximately 25 to 30%, with the remainder appearing as heat. This is, by the standards of biological systems and many engineered systems, remarkably good. The muscle is a molecular engine, and it is the engine of everything you do. The basic structural and functional unit of skeletal muscle is the sarcomere, a precisely organized arrangement of protein filaments that spans approximately 2 micrometers in the resting state and that shortens during contraction as interdigitating arrays of thin and thick filaments slide past each other in the mechanism known as the sliding filament theory. The sarcomere is one of those structures in biology that, when you understand it, makes the complexity of the body seem suddenly and almost uncomfortably ordered, as though the chaos of life is built on a foundation of molecular geometry that is almost crystalline in its regularity. A sarcomere is defined by its Z disks, the dense protein structures that form its boundaries, from which the thin filaments of actin extend inward toward the center. In the center of the sarcomere, the thick filaments of myosin are anchored by the M-line protein complex, their heads pointing outward in both directions toward the actin filaments at each end. The region of overlap between the actin and myosin filaments, the A-band, is visible as a darker zone in electron micrographs. In the region of pure actin at each end, the I-band, is lighter. The H-zone at the very center, containing only myosin without overlapping actin, disappears as the sarcomere shortens, because the actin filaments slide in from both ends, progressively filling the central zone. This sliding, first proposed by Andrew Huxley and Rolf Niederkirk, and simultaneously by Hugh Huxley and Gene Hansen, in their landmark papers of 1954, is the mechanism of muscle contraction. The filaments do not shorten. The actin and myosin themselves do not compress. They slide. The myosin heads engage the actin filament, pull it toward the center, release, reattach further along, and pull again. A ratcheting mechanism that, repeated millions of times per second across millions of sarcomeres in series within a single myofibril, produces the shortening of the fiber and ultimately the force that moves the world. The sarcomere is approximately 2 micrometers long in the resting muscle. Under the electron microscope, in a longitudinal section of skeletal muscle, you can see the repeating pattern of Z disks and A bands and I bands running along the length of every myofibril like the repeat of a molecular wallpaper pattern. The striated appearance of skeletal muscle, the reason it is called striated muscle, is this repeating sarcomere pattern, visible even at the light microscope level. The beautiful regularity of it is the regularity of a machine, which is exactly what it is. Calcium ions are the trigger of muscle contraction. Without a rise in intracellular calcium concentration, even a fully energized muscle with intact actin and myosin filaments cannot contract, and the machinery for releasing, Sequestering and cycling calcium within the muscle fiber is one of the most sophisticated intracellular signaling systems in the body. In the resting muscle fiber, the actin filament is blocked. Two regulatory proteins, tropomyosin, a long fibrous protein that runs along the groove of the actin helix, and troponin, a globular complex that sits at regular intervals along the tropomyosin, together maintain the actin in a state in which the myosin binding sites are obscured, preventing the myosin heads from attaching and generating force. The muscle is energized. ATP is present. Myosin heads are cocked and ready, but it cannot contract because the binding sites are covered. When a motor neuron fires and its action potential reaches the neuromuscular junction, acetylcholine is released into the synapse, depolarizes the muscle fiber membrane, and the resulting action potential spreads along the surface of the fiber and then dives inward along the T-tubules, invaginations of the surface membrane that penetrate into the interior of the fiber, bringing the electrical signal to within nanometers of the sarcoplasmic reticulum, the muscle's internal calcium store. The proximity of the T-tubule to the sarcoplasmic reticulum allows a protein coupling. The DHPR receptor in the T-tubule membrane interacting with the ryanodine receptor in the sarcoplasmic reticulum membrane that opens the calcium release channels in a flood. Calcium ions pour out of the sarcoplasmic reticulum into the cytoplasm of the fiber, and this rise in calcium concentration is the trigger. Calcium binds to troponin C, causing a conformational change in the troponin-tropomyosin complex that moves the tropomyosin out of the blocking position, exposing the myosin-binding sites on the actin. Myosin heads attach. The power stroke occurs. The sarcomeres shorten. The fiber contracts. When the motor neuron stops firing, the calcium pumps of the sarcoplasmic reticulum rapidly sequester the free calcium back into the reticulum. The tropomyosin returns to its blocking position. The myosin heads can no longer attach, and the fiber relaxes. Contraction is calcium. Relaxation is the removal of calcium. The entire complex event of muscle contraction is fundamentally a calcium-mediated molecular switch, set and reset by the electrical activity of the nervous system. Skeletal muscle fibers are among the largest cells in the human body, with the longest fibers in muscles like the sartorius reaching lengths of up to 30 centimeters, and they are multinucleated, containing hundreds or even thousands of nuclei per fiber, because no single nucleus could support the metabolic demands of a cell of this extraordinary size. The muscle fiber as a cell is a violation of the usual rules that govern cell size. Most cells in the body are measured in micrometers, tens to hundreds of micrometers in diameter, typically only a few micrometers in length. The cell nucleus, containing the genetic information and regulating the cell's gene expression, can service a cell of this size without difficulty. The longest muscle fibers of the sartorius, however, are some 30 centimeters long, 300 millimeters, 300,000 micrometers, and they are not thin. In a mature muscle fiber, The diameter ranges from 50 to 100 micrometers. A single nucleus, positioned at one end of such a fiber, could not possibly export mRNA and regulate the protein synthesis occurring at the distant ends of the cell in any meaningful time frame. The solution the body arrived at is the syncytium, the fusion of many mononucleate precursor cells into a single, multinucleate fiber. The satellite cells and myoblasts that fuse to form skeletal muscle fibers during development, and the satellite cells that fuse into existing fibers during repair and hypertrophy, contribute their nuclei to the growing fiber, each nucleus remaining responsible for its local region of cytoplasm, the myonuclear domain it governs. A large muscle fiber may contain several hundred to several thousand myonuclei, distributed along its length, each overseeing a territory of a few hundred micrometers. These nuclei are pushed to the periphery of the fiber, just beneath the cell membrane, as the interior is progressively filled with the dense packing of myofibrils. When you look at a cross-section of skeletal muscle in histology, the regular polygonal profiles of the fibers, each ringed with small dark dots at the periphery, those dots are the nuclei, the tiny governance centers of each local domain, maintaining the metabolic machinery that supports the contractile apparatus they surround. The neuromuscular junction, the specialized synapse where a motor neuron makes contact with a muscle fiber, is the largest and most thoroughly studied synapse in the body, and the precision of its signal transmission, ensuring that every action potential in the motor neuron reliably produces an action potential in the muscle fiber, is maintained by a remarkable excess of neurotransmitter. The neuromuscular junction does not operate on the margins of adequacy. It operates with a safety factor so large that the junction can still function reliably after losing the majority of its acetylcholine receptors, a reserve that is, in health, never called upon but that provides protection against the kinds of partial disruption that occur in disease. When a motor neuron action potential reaches the presynaptic terminal, voltage-gated calcium channels in the terminal membrane open, calcium enters, and the vesicular machinery responds by fusing approximately 50 to 100 acetylcholine-containing vesicles with the presynaptic membrane, releasing their contents, approximately 5 to 10,000 molecules of acetylcholine per vesicle, into the synaptic cleft. This acetylcholine diffuses rapidly across the synaptic cleft and binds to the nicotinic acetylcholine receptors densely packed in the folds of the postsynaptic membrane, the motor end plate. The binding opens the receptor ion channels. Sodium and potassium ions flow. The end plate depolarizes, and this end plate potential is large enough, under normal circumstances, with all receptors intact, to reliably trigger an action potential in the muscle fiber. Large enough, in fact, to trigger an action potential even if 70 or 80% of the receptors were non-functional. This is the safety factor, the ratio of the actual end plate potential to the threshold required for an action potential, which in healthy human muscle is approximately 3 to 5. It is this safety factor that is eroded in myasthenia gravis, the autoimmune condition in which antibodies target and destroy the acetylcholine receptors of the neuromuscular junction. As the receptor density falls, the safety factor falls with it, until eventually the end plate potential can no longer reliably trigger a muscle action potential, and the characteristic fatigable weakness of myasthenia gravis appears. The normal junction is so far above threshold that this erosion takes time and considerable receptor loss before it becomes symptomatic. The junction is engineered to be reliable. It is reliable to a degree that only becomes apparent when it begins to fail. Muscle fibers are classified into types based on their speed of contraction, their metabolic characteristics, and their fatigue resistance. The classic division into type 1 slow-twitch oxidative fibers and type 2 fast-twitch fibers, with the fast-twitch category further subdivided into type IA and type I9, represents a spectrum of mechanical and metabolic properties that reflects the diverse demands placed on different muscles in different activities. The three fiber types are not equally distributed across all muscles or in all individuals, and the particular mixture of fiber types in a given muscle tells you something about the habitual function of that muscle and, in athletes, something about the training history and genetic endowment of the person who owns it. Type 1 fibers, slow twitch, oxidative, are the endurance fibers. They contract relatively slowly, generating moderate force, but they are extraordinarily resistant to fatigue because they generate ATP almost exclusively through oxidative phosphorylation, which is energetically efficient and produces almost unlimited ATP as long as oxygen and fuel are available. They are densely packed with mitochondria, richly supplied with capillaries, and contain large amounts of myoglobin, the muscle's oxygen-binding protein, responsible for the deep red color of slow-twitch muscle. The diaphragm, the postural muscles of the spine, and the soleus of the calf, muscles that must work continuously, without rest, for a lifetime, are predominantly type 1. Type I9 fibers, the fastest and most powerful, are at the other extreme. They contract rapidly and generate large force, but they rely primarily on glycolytic metabolism, which is fast but not sustainable, and they fatigue rapidly. Sprinters have a high proportion of type I9 fibers in their leg muscles. Endurance runners have a high proportion of type 1. Type IA fibers sit between the extremes, faster than type 1, more fatigue-resistant than type I9, with a mixed metabolic profile that makes them well-suited to the moderate intensity, moderate duration activities that characterize much of daily life. The relative proportions of these fiber types are partly genetically determined. Elite sprinters and elite endurance runners are born, in part, with the fiber type distribution their sports require, and partly responsive to training, with endurance training promoting the transformation of type I9 toward type IA. The muscle, in other words, adapts its cellular composition to the demands placed on it. It becomes what you use it to be. The force a muscle can generate is not constant but varies with its length, a relationship called the length-tension relationship, such that the muscle produces maximum force at a specific optimal length and less force at lengths either shorter or longer than this optimum. because of the way the overlap between actin and myosin filaments changes with sarcomere length. The length-tension relationship is one of the fundamental properties of muscle, and understanding it requires thinking about what is happening at the level of individual sarcomeres as the muscle changes length. At very short sarcomere lengths, the actin filaments from the two Z discs of the same sarcomere begin to overlap each other in the center, and the myosin filaments come into contact with the Z discs. Both of these geometric collisions interfere with cross-bridge formation and force generation. The actin from one side is competing with the actin from the other side for myosin binding sites, and the myosin heads near the tips of the thick filament are being blocked by the Z disc. Force production falls steeply at very short lengths. At very long sarcomere lengths, the actin and myosin filaments no longer overlap. The thin filaments have been pulled out from the space between the thick filaments, and myosin heads have no actin to attach to. No overlap, no cross bridges, no force. Force is zero at lengths where there is no filament overlap. Between these extremes, force is proportional to the degree of filament overlap, reaching a maximum at the length where every myosin head on the thick filament has an available actin binding site, the optimal overlap condition. In vivo, the joint geometry and the passive restraints on limb position keep most muscles operating near their optimal length for most activities, which is a consequence of the fact that the musculoskeletal system evolved to position muscles at their most advantageous working lengths. But the relationship explains many things about the variation in muscle strength with joint angle. Why a bicep curl is weakest at full elbow extension and full elbow flexion and strongest at the mid-range. Why a deep squat requires more effort than a partial one in ways that the load alone does not fully explain. The force a muscle produces is not simply a property of the muscle. It is a property of the muscle at a particular length, and length is determined by joint angle. And joint angle is something you control. The diaphragm, the dome-shaped sheet of muscle and tendon that separates the thoracic and abdominal cavities, is the primary muscle of breathing, responsible for approximately 60 to 80% of the work of quiet inspiration, and it contracts roughly 20,000 times per day without rest, making it possibly the most active voluntary muscle in the body. The diaphragm is architecturally unlike most skeletal muscles. It is a flat, sheet-like structure rather than a fusiform or pinnate muscle mass, and it is arranged as a dome, convex upward into the thoracic cavity when at rest, with a central tendon at its apex and muscle fibers radiating outward and downward from this central tendon to their attachments around the periphery of the lower thorax. Anteriorly, the muscle fibers attach to the lower sternum and the xiphoid process. Laterally and posteriorly, they attach to the inner surfaces of the lower 6 ribs and their costal cartilages. Most posteriorly, the crural fibers, the diaphragm's legs, descend to attach to the upper lumbar vertebrae and the arcuate ligaments. When the diaphragm contracts, the dome flattens and descends, increasing the vertical dimension of the thoracic cavity and driving down the abdominal contents below. This descent in the face of the relatively incompressible abdominal viscera, The intestines and liver and stomach cannot be compressed, only displaced, causes the anterior abdominal wall to move outward, which is the visible movement of the belly breathing you may have learned about in relaxation practice. The descent of the diaphragm reduces the pressure inside the thoracic cavity below atmospheric pressure, and air flows in through the airways in response to this pressure gradient. Exhalation and quiet breathing is largely passive. The diaphragm relaxes, the lungs' elastic recoil drives the air out, and the dome ascends. The remarkable thing about the diaphragm is not just its mechanical action but its endurance. It has contracted approximately 20,000 times today. It contracted approximately 20,000 times yesterday. It will do so again tomorrow. In 70 years of life, the diaphragm makes somewhere in the vicinity of 500 million contractions. No skeletal muscle comes close to this. Right now, as you lie here, it is working, as it always has. The heart muscle, cardiac muscle, or myocardium, is fundamentally different from both skeletal and smooth muscle in its structure and in its electrical properties, most significantly in its ability to generate its own electrical impulses through pacemaker cells, making the heart a muscle that does not require instruction from the nervous system to beat. The distinction between cardiac and skeletal muscle goes deeper than mere structural differences in the arrangement of the sarcomeres, which in cardiac muscle are also present and organized in the same regular pattern. Cardiac muscle cells, cardiomyocytes, are shorter and branched rather than the long, unbranched cylinders of skeletal muscle. They are mononucleated or binucleate rather than multinucleated. They are connected to their neighbors by structures called intercalated discs, specialized cell junctions that contain gap junctions allowing the direct passage of ions between adjacent cells and anchoring junctions that mechanically couple adjacent cells so they contract as a syncytium. The gap junctions of the intercalated discs allow the action potential to spread from cell to cell across the entire myocardium as a single wave, ensuring that all the cells of each chamber contract together rather than in the disorganized, uncoordinated way that would be ineffective for pumping. But the most important difference is the ability of certain cardiac cells to act as pacemakers, to depolarize spontaneously, without any external stimulus, generating the electrical impulse that drives the heartbeat. The sinoatrial node, located in the wall of the right atrium, contains the cells with the fastest intrinsic rate of spontaneous depolarization, typically 60 to 100 times per minute in the resting adult. And this rate sets the heart rate by depolarizing the rest of the myocardium faster than any other potential pacemaker can do so on its own. The nervous system modulates this rate, the vagus nerve slowing it, the sympathetic system accelerating it, but it does not initiate it. The heart beats because the heart has decided to beat. The pacemaker cells do not wait for permission. They have been generating their spontaneous impulses since early in fetal development, long before the nervous system reached its current sophistication, and they will continue as long as the cells live. Tonight, as you rest, Your heart is doing this approximately 60 times per minute, each impulse spreading as a wave of electrical activity across the 4 chambers in a precisely timed sequence. Each wave followed by the mechanical contraction that pushes the blood through the circulation. 60 times per minute, 3,600 times per hour, 86,400 times per day. The cardiac muscle does not know how to stop. Smooth muscle, the non-striated, involuntary muscle that lines the blood vessels, the digestive tract, the respiratory airways, the uterus, and many other visceral structures, is controlled by the autonomic nervous system and by local chemical signals, and its distinctive properties include the ability to maintain sustained contraction at very low energy cost, a feature called the latch state. Smooth muscle is the invisible muscle, the muscle of the interior, the muscle that operates entirely beyond voluntary control, the muscle that you have never consciously commanded and yet which has been governing the inner workings of your body every moment of your life. Its cells are small and spindle-shaped, each with a single nucleus, each containing actin and myosin filaments but arranged not in the regular sarcomere pattern of striated muscle but in a more oblique, less organized configuration that allows the cell to shorten by a greater fraction of its resting length than a skeletal muscle sarcomere can. Smooth muscle has no T-tubule system and no sarcoplasmic reticulum as elaborate as skeletal muscle. Its calcium handling is different. Calcium enters from the extracellular space as well as from internal stores, and it activates contraction through a different molecular mechanism, via calmodulin and myosin light chain kinase rather than troponin and tropomyosin. The result of this different calcium activation mechanism is a slower onset of contraction. Smooth muscle does not generate force as rapidly as skeletal muscle, but a potentially much more sustained one. The latch state is smooth muscle's most remarkable adaptation, a condition in which cross bridges form and remain attached for a very long time without detaching and reattaching in the rapid cycling that characterizes skeletal muscle contraction. Latch state bridges generate force but consume very little ATP, because most of the energy cost of skeletal muscle contraction comes from the detachment and recycling steps. A blood vessel maintaining its tone against the pressure of the blood within it, hour after hour, day after day, does so using a fraction of the energy that an equivalent skeletal muscle contraction would require. Smooth muscle tires very little. It simply holds. The motor unit, the combination of a single motor neuron and all the muscle fibers it innervates, is the fundamental unit of motor control, and the progressive recruitment of motor units in order from smallest to largest as force requirements increase, known as Henneman's size principle, is one of the most elegant and well-established principles in motor neuroscience. A motor unit is not a structure that you can see under a microscope. It is a functional unit, defined by connectivity rather than anatomy. The motor neuron cell body lives in the ventral horn of the spinal cord. Its axon travels in the peripheral nerve to the muscle, where it branches at the neuromuscular junction to innervate a specific number of muscle fibers scattered throughout the muscle. The size of the motor unit, the number of fibers it innervates, varies enormously depending on the muscle's function. In the extraocular muscles that move the eye, a single motor neuron may innervate only 3 to 5 muscle fibers. Fine-grained control, the ability to make tiny adjustments in gaze direction, requires that each small increment of neural activity produce only a tiny increment of force. In the large muscles of the lower limb, the quadriceps, the gastrocnemius, a single motor neuron may innervate several hundred to over 1,000 muscle fibers. These muscles need to generate large forces and fine gradations within the range of large forces, but they do not need the same extraordinary resolution of fine motor control. Henneman's size principle states that motor units are recruited in a fixed order, from smallest to largest, as the force demand increases. Small motor units, with small motor neurons, innervating few fibers, predominantly type 1 slow twitch, are recruited first, at low force levels. As force demand increases, progressively larger motor units, with larger motor neurons, innervating more fibers, increasingly type 2 fast twitch, are added. This orderly, size-based recruitment has the consequence of automatically matching the type of motor unit to the intensity of the task. Low intensity, sustained activities use predominantly small, fatigue-resistant type 1 units. High intensity, brief efforts bring in the large, powerful, fatigable type 2 units only when needed. The principle is elegant because it requires no central computation to implement. It arises automatically from the fact that smaller motor neurons have higher input resistance and therefore respond to a given level of synaptic input with a greater change in membrane potential, reaching their firing threshold before larger neurons. The size principle is a structural consequence, not a programmed strategy. The nervous system stumbled, through evolution, onto a motor control principle that a motor engineer might have designed deliberately. Muscle hypertrophy, the increase in muscle size in response to resistance training, occurs primarily through an increase in the size of individual muscle fibers rather than an increase in their number, and the molecular mechanism involves the activation of satellite cells and the stimulation of protein synthesis through the mTOR signaling pathway. The question of how exercise makes muscle bigger is one that has attracted enormous scientific attention, partly because of the medical and athletic implications of the answer and partly because the answer turns out to be more complex and more interesting than early models suggested. The dominant early model, that resistance training adds new sarcomeres, thickening the fibers, is essentially correct. But the process by which this happens involves a cellular drama that operates over hours and days following a single exercise bout. Intense resistance exercise damages the muscle fibers. The high forces of contraction and particularly the eccentric lengthening contractions produce microdamage to the sarcomere structure and the sarcolemmal membrane, which might seem like a poor basis for building a stronger muscle. But this damage is the stimulus. The damaged fibers release cytokines, inflammatory signals, that recruit macrophages to the muscle, and the macrophages in turn activate the satellite cells, the muscle's resident stem cell population that ordinarily lie quiescent in a shallow depression between the fiber membrane and its basement membrane. Activated satellite cells divide, and a proportion of their daughters fuse into the existing fiber, donating their nuclei and contributing to the increased myonuclear domain required to support protein synthesis in a larger cell. Meanwhile, the mechanical and metabolic stress of the exercise activates the mTOR pathway, mechanistic target of rapamycin, a central regulator of cell growth, which upregulates ribosome production and the translation of the mRNAs encoding the contractile proteins, actin, myosin, titin, tropomyosin, troponin. More ribosomes producing more contractile protein means more sarcomeres added in parallel, thickening the myofibrils, expanding the cross-sectional area of the fiber, and ultimately enlarging the muscle. The process is not fast. Measurable hypertrophy from a single exercise bout takes days to manifest, and meaningful changes in muscle size require weeks to months of consistent training. The muscle grows reluctantly, in response to a demand it has concluded is serious and repeated. It does not waste resources building capacity that the organism has shown no commitment to using. The force-velocity relationship of muscle describes the inverse relationship between the velocity of muscle shortening and the force the muscle can generate. A muscle shortening rapidly produces less force than one shortening slowly, and the maximum force is produced at zero velocity. And this relationship has profound implications for athletic performance and for the design of movement. The force-velocity relationship was described quantitatively by Archibald Hill in 1938, in one of the most influential papers in muscle physiology, and the hyperbolic curve that relates force to velocity, the Hill equation, remains one of the most robust quantitative relationships in the field. The relationship has a simple physical basis. At high shortening velocities, the myosin heads cannot complete their attachment, power stroke, and detachment cycle fast enough to maintain high crossbridge numbers. At the moment of detachment, some heads that would normally reattach immediately are instead swept forward by the rapid movement of the actin filament before they can find a new binding site, reducing the instantaneous number of attached crossbridges and therefore the instantaneous force. The faster the shortening, the more this problem is compounded, and the lower the force. At very high velocities, the maximum shortening velocity, Vmax, so few crossbridges are attached at any instant that force approaches zero. Conversely, the maximum force, the isometric force, the force at zero velocity, is produced when all available crossbridges can complete their cycle without the disruption of filament movement. The force-velocity relationship extends to the eccentric side of zero velocity, when the muscle is being lengthened while active. And here something interesting happens. The force is greater than the isometric maximum, sometimes substantially so. Eccentric contractions, in which the muscle generates force while being stretched, can produce forces 20 to 50% greater than the maximum isometric force. This is why lowering a heavy weight, the eccentric phase of a bicep curl, can be performed with heavier loads than the lifting phase, and why eccentric training is particularly effective for building muscle. It is also why muscle soreness after exercise, delayed onset muscle soreness, is predominantly an eccentric phenomenon, because the high forces of eccentric contractions are particularly damaging to the sarcomere structure. The muscle produces its greatest force when it is being forced to lengthen. There is something quietly ironic in that the most powerful state of muscle is the one in which it is losing the battle. The pennation angle of a muscle, the angle at which its fibers are arranged relative to the line of action of the muscle, determines the trade-off between the force the muscle can generate and the range of motion it can produce, with pennate muscles sacrificing range of motion for increased force-generating capacity. Most people, when they imagine a muscle, picture a fusiform structure, a spindle shape, thickest in the middle, tapering to tendons at each end, with the fibers running parallel to the long axis of the muscle. This architecture, parallel fibers along the line of pull, maximizes the range of motion the muscle can produce, because the full shortening of each fiber is transmitted directly as shortening of the whole muscle. The biceps brachii is a reasonably good example of this parallel architecture. But many muscles in the body are pennate. Their fibers run at an angle to the line of pull, attaching to a central tendon or aponeurosis in an oblique arrangement reminiscent of the barbs of a feather. The gastrocnemius, the tibialis anterior, the rectus femoris, the deltoid, all are pennate to varying degrees. Pennation reduces the effective range of motion the muscle can produce, because the fiber shortening is transmitted to the line of pull not at full value but multiplied by the cosine of the pennation angle, the component of the fiber's shortening that acts along the direction of the tendon. For typical pennation angles of 15 to 30 degrees, this loss of effective shortening is relatively modest. But pennation confers a significant advantage in force production by allowing more fibers to be packed into the same volume of muscle. The fibers are shorter and more oblique, so more of them fit. Pennate muscles have a higher physiological cross-sectional area relative to their anatomical size, and since force is proportional to cross-sectional area, they can generate more force. The quadriceps, enormously powerful, pennate, relatively short-fibered, exemplifies this design. Its force-generating capacity per unit muscle volume is greater than a parallel fibered equivalent would be, at the cost of a somewhat reduced range of motion. The architecture of each muscle is the solution to the specific trade-off between force and motion that the muscle's functional role demands. Muscle fatigue, the decline in force-generating capacity during sustained or repeated contractions, is not a single phenomenon but a complex of multiple mechanisms operating at different sites in the excitation-contraction chain, from the central nervous system to the individual myofilaments, and the relative contribution of each mechanism depends on the type and duration of the exercise. The popular conception of muscle fatigue, that it is caused by lactic acid accumulation, is one of the most persistent misconceptions in exercise physiology, and its persistence is a testimony to the durability of ideas that are wrong in an interesting way. Lactic acid production does occur during high-intensity exercise, and the associated acidosis was long assumed to be the primary cause of fatigue. But subsequent research has complicated this picture substantially. During brief, high-intensity exercise, the accumulation of inorganic phosphate from the breakdown of phosphocreatine and from cross-bridge cycling now appears to be more important than acidosis in reducing force production. Inorganic phosphate directly inhibits the power stroke of the myosin head and reduces the force per cross-bridge. During prolonged exercise, glycogen depletion within the muscle fibers reduces the availability of the fuel that sustains calcium pumping and cross-bridge cycling, impairing the muscle's ability to maintain force. Calcium leakage from damaged sarcoplasmic reticulum reduces the calcium available for the next stimulus. And the central nervous system plays a role that has been increasingly recognized. The reduction in motor drive from the brain, sometimes called central fatigue, is a real and significant contributor to the performance decrements of prolonged exercise, involving changes in neurotransmitter systems, accumulation of serotonin and other signals that modulate the perceived effort of exercise. At the neuromuscular junction, high-frequency stimulation can deplete the pool of renally releasable acetylcholine vesicles, reducing the safety factor of transmission. At the muscle membrane, the accumulation of potassium in the T-tubule system can impair the propagation of the action potential into the fiber interior. Fatigue is not one thing. It is the sum of many things, each dominant at different times and intensities, each serving in its way the function that fatigue always ultimately serves, protecting the muscle from damage that unmodulated force generation would cause. The tension a muscle can develop is determined not just by the number of cross bridges but by the rate of cross-bridge cycling, and the calcium sensitivity of the contractile apparatus can be modulated by post-translational modifications of the regulatory proteins, providing a mechanism for tuning muscle performance independent of the calcium signal itself. The regulation of muscle contraction has long been understood in terms of the calcium-troponin-tropomyosin switch. Calcium rises, troponin changes conformation, tropomyosin moves, actin is exposed, myosin attaches, force is generated. This is the primary regulatory mechanism, and it is real and important. But the performance of the muscle given a particular calcium signal can be further modulated by a variety of post-translational modifications of the contractile proteins. Chemical changes made to the proteins after they have been synthesized, which alter their functional properties without changing the genes that encode them or the sequences of the proteins themselves. Phosphorylation. The addition of a phosphate group by a protein kinase is the most important of these modifications in the context of muscle regulation. Phosphorylation of the myosin regulatory light chains, small proteins associated with the myosin head, has been shown to increase the calcium sensitivity of the contractile apparatus in smooth muscle and to increase the rate of cross-bridge cycling in fast skeletal muscle. Phosphorylation of troponin I, one of the three subunits of the troponin complex, by protein kinase A in response to beta-adrenergic stimulation decreases the calcium affinity of troponin C, speeding the relaxation of the muscle after each contraction. This is part of the mechanism by which adrenaline makes the heart beat both faster and more forcefully. It phosphorylates both myosin light chains and troponin I, increasing force development and accelerating relaxation, allowing the heart to fill and eject at the faster rates demanded by exercise. These regulatory modifications allow the performance of the muscle to be tuned by the hormonal and neural environment beyond what the calcium signal alone can achieve. An additional layer of control sitting on top of the primary calcium switch, adjusting the sensitivity and the kinetics of the response to the moment's specific demands. The passive elastic properties of muscle, provided primarily by the giant protein titin, which spans the full half sarcomere from the Z disc to the M line and functions as a molecular spring, play important roles in muscle mechanics, storing energy during stretch and returning it during subsequent shortening, and contributing to the stability of the sarcomere during eccentric contractions. Titin is the largest protein in the human body by molecular weight. A single titin molecule spans approximately 1 micrometer, roughly half the resting sarcomere length, and its molecular weight of approximately 3 to 4 million Daltons dwarfs all other proteins. It is anchored at one end to the Z disc and at the other to the M line, where it interacts with the thick filament. In the A-band region, the region of the sarcomere occupied by the thick filament, titin is inextensible, bound tightly to the myosin. In the I-band region, the region of pure actin between the end of the thick filament and the Z disc, titin is extensible, acting as a molecular spring. The spring-like region of titin contains two structurally distinct domains, immunoglobulin-like repeating domains that unfold sequentially as the sarcomere is stretched, and a unique sequence called the PEVK domain, named for its high content of the amino acids proline, glutamate, valine, and lysine, that extends more readily and contributes the initial low stiffness response to stretch. Together, these domains make titin a nonlinear spring, stiff at long sarcomere lengths, softer at shorter lengths, whose mechanical properties are precisely tuned to the working range of the sarcomere. During eccentric contractions, active stretching, titin is also activated by calcium binding to store additional elastic energy, increasing its stiffness in a calcium-dependent manner. This calcium-dependent stiffening of titin is now understood to be a significant contributor to the force enhancement above the isometric maximum that is characteristic of eccentric contractions. Without titin, the sarcomere would be mechanically unstable. The thick and thin filaments would not return to their correct relative positions after being displaced. Titin is not just a spring. It is the sarcomere's structural memory, the element that ensures that after every contraction, every stretch, every distortion, the sarcomere returns to its proper form. The muscles of the pelvic floor form a hammock of interlocking muscle and connective tissue that supports the pelvic viscera against the downward force of intra-abdominal pressure, and their continuous low-level activation, largely below conscious awareness, is one of the most demanding and least recognized aspects of postural muscle function. The pelvic floor is a structure that, until it fails, most people are only vaguely aware of possessing. It consists of several overlapping layers of muscle, primarily the levator ani group, comprising the pubococcygeus, puborectalis, and iliococcygeus muscles, and the coccygeus, that attach anteriorly to the pubic bones, laterally to the ischial spines and the obturator internus fascia, and posteriorly to the coccyx and sacrum, forming a broad muscular platform that closes the bottom of the pelvic cavity. Through this platform pass the urethra, the rectum, and in females the vagina, each surrounded by loops of levator ani muscle that provide both support and a sphincter-like function. The pelvic floor muscles are skeletal muscles, under both voluntary and reflex control, and they respond to increases in intra-abdominal pressure, coughing, sneezing, laughing, lifting, with a reflex precontraction that stiffens the pelvic floor a fraction of a second before the pressure increase arrives. This anticipatory contraction, coordinated with similar anticipatory contractions in the transversus abdominis and the diaphragm, is part of the core stabilization system that protects the lumbopelvic region from the forced surges of movement and exertion. In upright posture, the pelvic floor is continuously active at a low level, supporting the weight of the pelvic organs against gravity. This continuous activity is metabolically modest. The fibers involved are predominantly type 1 slow twitch, ideally suited to sustained low-level contractions, but it is mechanically essential. The failure of this continuous support, in the context of connective tissue weakening or neural damage, produces the prolapse and incontinence that affect a significant proportion of the population over a lifetime. The pelvic floor is doing its quiet work right now, even as you lie horizontal and the gravitational demand is minimized. Even in rest, some muscles never quite let go. Myosin is not a single protein but a large family of motor proteins with diverse functions throughout the cell, and the specific isoform of myosin expressed in a muscle fiber, its myosin heavy chain, is the primary determinant of its fiber type and its speed of contraction, with different myosin heavy chain isoforms having different ATPase rates that determine how fast the sarcomere cycles. The myosin of skeletal muscle, myosin II, the conventional muscle myosin, is a hexameric protein consisting of 2 heavy chains and 2 pairs of light chains, assembled into a molecule shaped somewhat like a golf club, a long coiled coil tail formed by the intertwining of the 2 heavy chains, and 2 globular heads at one end, each formed from the N-terminal portion of 1 heavy chain and associated light chains. The heads contain the ATP binding and actin binding sites, the molecular machinery of the power stroke. The tails of many myosin molecules associate with each other through their coiled coil regions to form the bipolar thick filaments of the sarcomere. 600 myosin molecules per thick filament, their heads pointing outward from the central bare zone in each direction, like the bristles of a molecular bottle brush. The speed at which a muscle fiber contracts, its unloaded shortening velocity, its Vmax, is determined primarily by the specific myosin heavy chain isoform it expresses, which determines the ATPase rate of the myosin head. The faster the ATPase rate, the faster ATP is hydrolyzed, the faster each power stroke cycle is completed, and the faster the sarcomere can shorten. Type 1 fibers express the slow myosin heavy chain isoform, MHCI, or MHC beta in cardiac muscle, with a slow ATPase rate and a low maximum shortening velocity. Type I9 fibers express MHCI9, with the fastest ATPase rate and the highest maximum shortening velocity. MHCIA occupies the intermediate position. Each isoform is encoded by a separate gene, and the expression of these genes is regulated by neural activity, mechanical loading, and hormonal signals, which is why the fiber type composition of a muscle can shift with changes in activity level or training. When you train for endurance, your muscles increase the relative expression of the slower myosin heavy chain genes. When you train for power, the balance shifts toward the faster isoforms. The muscle, at the level of gene expression, is listening to what you ask of it. The muscles of the eye, the 6 extraocular muscles that position the globe in the orbit, are the fastest skeletal muscles in the body and contain muscle fibers unlike any found elsewhere, including multiply innervated fibers that grade their tension tonically without action potentials, providing a unique mechanism for fine control of eye position. The extraocular muscles perform movements of a speed and precision that place them in a different category from other skeletal muscles. The saccade, the rapid, ballistic eye movement that shifts the gaze from one point to another, is the fastest movement the human body can produce, reaching angular velocities of up to 700 degrees per second and completing in as little as 20 milliseconds. To generate this speed, the extraocular muscles contain fast twitch muscle fibers expressing the fastest myosin heavy chain isoforms, with cross-bridge cycling rates that exceed those in any other muscle. But speed is only part of the story. The eyes must also hold gaze, maintain a precise position against perturbations, with the stability measured in fractions of a degree, for sustained periods. For this fine positional control, the extraocular muscles contain a specialized fiber type found nowhere else in the human body, the multiply innervated, non-twitch fiber. These fibers have multiple neuromuscular junctions distributed along their length, rather than the single end plate of the usual twitch fiber. They do not generate action potentials. Instead, the end plate potentials from multiple junctions summate across the fiber, producing a graded, tonic depolarization that activates the contractile apparatus at a level proportional to the motor neuron firing rate. This produces a smooth, graded, continuously variable tension, exactly what is needed for precise position control, without the all-or-none character of the twitch fiber. The extraocular muscles thus contain, side by side, the two most different muscle fiber types in the human body, the fastest twitch fibers for saccades and the unique non-twitch fibers for gaze maintenance. The same muscle, Two different solutions to two different problems, expressed simultaneously in the fibers that move your eyes. The elastic tendons that connect muscles to bones are not passive cables but energy-storing springs that play an active role in locomotion, with the Achilles tendon alone estimated to contribute sufficient elastic energy return to reduce the metabolic cost of running by approximately 50% compared to what running without a compliant tendon would require. The tendon is a simple cable, a passive, inextensible link transmitting force from muscle to bone, is an adequate model for some tendons in some situations but is profoundly inadequate as a general description of the role tendons play in locomotion. Tendons are compliant. They stretch under load, storing elastic strain energy, and they recoil when the load is released, returning a large fraction of the stored energy. The Achilles tendon is the clearest and most extensively studied example of this elastic function. During the stance phase of running, the ankle dorsiflexes under the body's weight, stretching the Achilles tendon and the aponeurosis of the gastrosoleus complex. The tendon strain, the fractional elongation under load, reaches approximately 6% at the peak load of running, storing elastic strain energy. As the ankle plantar flexes during push-off, this stored energy is released, supplementing the contractile force of the calf muscles and reducing the metabolic demand on them. The efficiency of this energy storage and return, typically 90 to 93%, meaning only 7 to 10% of the stored energy is lost as heat, makes the tendon one of the most efficient springs known. Calculations suggest that if the Achilles tendon were replaced by an inextensible cable, the metabolic cost of running would approximately double, because the calf muscles would have to generate all the force of push-off through contractile work rather than elastic recoil. A similar function has been demonstrated for the arch of the foot, for the plantar fascia, and for the patellar tendon. The compliant spring of the tendon is so central to the energy economy of bipedal running that some researchers argue it was the critical innovation in the evolution of endurance running in the human lineage. The ability to run long distances at a sustainable metabolic cost being inseparable from the development of long, compliant tendons that could store and return energy with each stride. The connective tissue framework of muscle, the epimysium surrounding the whole muscle, the perimysium surrounding each fascicle, and the endomysium surrounding each individual fiber, is not merely structural packaging but plays active roles in transmitting force, distributing mechanical load, and providing the scaffold for satellite cell activation during repair. The endomysium, the thin layer of connective tissue that surrounds each individual muscle fiber, is in direct contact with the basal lamina of the fiber and with the satellite cells that lie between the basal lamina and the sarcolemma. The fibrous proteins of the endomysium, primarily types I and III collagen, form a fine mesh that transmits the lateral forces generated by the contractile apparatus in the fiber to the adjacent fibers, through the surrounding connective tissue, and ultimately to the tendon. This lateral force transmission, sometimes called the shunt pathway for force, in contrast to the direct longitudinal pathway from fiber to tendon, is important in muscles where not all fibers run the full length of the muscle and therefore cannot attach directly to both the origin and insertion tendons. It allows the force of a short fiber to be transmitted into the connective tissue network and thence to the tendon, even without the fiber itself having a direct tendinous attachment at both ends. The perimysium, surrounding fascicles, is thicker and more complex than the endomysium and contains a higher proportion of type I collagen, providing greater stiffness. It also contains the blood vessels and nerves that supply the muscle, running between the fascicles before branching into the endomysial space. The epimysium, the outermost layer, merges with the connective tissue of adjacent structures, the deep fascia, the tendons, the periosteum of adjacent bones, creating a continuity of connective tissue that distributes mechanical forces throughout the musculoskeletal system. This facial continuity is part of what allows the body to be understood not just as a collection of individual muscles but as a mechanically integrated system in which forces flow through connective tissue networks across large distances. The mechanism by which motor neurons are recruited and their firing rates modulated to produce a smoothly graded increase in muscle force. The two components of the voluntary force control system are regulated by complex inputs from the cerebral cortex, the cerebellum, the basal ganglia, and multiple brainstem and spinal cord circuits, making voluntary movement one of the most computationally demanding tasks the brain performs. The apparent simplicity of picking up a cup of tea conceals a computational achievement of daunting complexity. The movement requires the brain to specify the desired outcome. The hand at the cup's location, closed around the handle with appropriate force, the elbow angle and shoulder orientation placed in their required configuration, and then to solve the inverse dynamics problem, given the desired outcome. What pattern of muscle forces, applied through the linkages of the skeleton and distributed across the joints, will produce that outcome? This inverse dynamics problem has no unique mathematical solution for a system as redundant as the human musculoskeletal apparatus. The shoulder alone has roughly 17 muscles spanning it, any combination of which can produce a given shoulder movement, and the optimal combination must be computed anew for each movement. The cerebral cortex provides the highest level command, the intention and the spatial planning. The basal ganglia modulate the selection and sequencing of movement programs. The cerebellum receives a copy of the intended movement and a continuous stream of sensory feedback, computing the difference between what was intended and what is actually happening, and generating corrective signals that are fed back into the motor system to minimize error. The spinal cord integrates all of these descending signals with the ascending sensory information from muscle spindles, Golgi tendon organs, joint receptors, and skin receptors, producing the final motor output to the alpha motor neurons that drive the muscle fibers. The motor cortex and its descending corticospinal tract are the most direct command pathway, but they operate as part of a system in which every level of the neuraxis contributes. The movement you make picking up a cup of tea is, in neural terms, a coordinated feat of the entire brain and spinal cord, carried off in a fraction of a second, executed so automatically that it requires no conscious effort beyond the initial decision to reach. The muscles are the final output of a computation that makes any human-designed robotic system look rudimentary. Muscle spindles, the sensory organs embedded within skeletal muscles that detect muscle length and the rate of change of length, provide the brain with continuous information about the state of the musculoskeletal system, and their role in proprioception makes them one of the most important sensory organs in the body, despite being essentially invisible in everyday experience. The muscle spindle is a remarkable intramuscular sensory organ, buried within the muscle belly, oriented parallel to the regular muscle fibers, and dedicated entirely to the measurement of two things: the length of the muscle and how fast that length is changing. Each spindle is a small, encapsulated structure about 5 to 10 millimeters long, containing a central fluid-filled space surrounded by a capsule of connective tissue, within which lie a small number of specialized modified muscle fibers, the intrafusal fibers, so called to distinguish them from the extrafusal fibers that make up the bulk of the muscle. The intrafusal fibers are innervated at their ends by small gamma motor neurons, which can adjust their tension independently of the main muscle contraction. This gamma innervation is what allows the spindle to remain sensitive across the full range of muscle lengths. If the spindle were rigidly parallel to the extrafusal fibers, it would go slack and become insensitive whenever the main muscle shortened. The gamma innervation takes up the slack, keeping the intrafusal fibers under tension and the spindle sensitive regardless of the overall muscle length. The spindle's sensory output comes from the primary and secondary afferent endings wrapped around the central equatorial region of the intrafusal fibers. The primary IA afferents are particularly sensitive to the rate of change of length. They fire rapidly during the dynamic phase of a stretch and less rapidly when the stretch has reached a steady state. The secondary two afferents are primarily sensitive to the steady state length. Together, these afferents provide the central nervous system with a continuous, accurate report of muscle length and velocity, information that is essential for the execution of skilled movement and for the maintenance of posture and balance. The stretch reflex, the knee-jerk reflex tapped by the doctor's hammer, is the simplest expression of the spindle's function. A stretch of the muscle activates the IA afferents, which synapse monosynaptically on the alpha motor neurons of the same muscle, driving a contraction that resists the stretch. Delayed onset muscle soreness, the familiar muscle ache that peaks 24 to 72 hours after unaccustomed exercise, particularly exercise involving eccentric contractions, is not caused by lactic acid accumulation but by a complex cascade of inflammatory and repair processes triggered by mechanical damage to the sarcomere structure. The lactic acid theory of delayed onset muscle soreness has persisted in popular understanding long after exercise science has moved on, partly because lactic acid is a vivid and comprehensible culprit and partly because the burning sensation during intense exercise has been conflated with the soreness that follows it. But the two phenomena are completely distinct. The burning during intense exercise is related to metabolic acidosis and potassium accumulation and resolves within minutes of stopping. Delayed onset muscle soreness begins hours after exercise, typically 6 to 12 hours after, and peaks at 24 to 72 hours. At the time of its onset, lactic acid levels in the muscle have been normal for hours. The actual mechanism begins with the structural disruption of the sarcomere during intense, particularly eccentric exercise. The high forces of eccentric contractions produce mechanical disturbances to the Z-discs, the boundaries of the sarcomere, which can tear or stream, and to the sarcolemma, which can develop small tears that allow the normally intracellular enzyme creatine kinase to leak into the bloodstream. This structural damage triggers an inflammatory response. Prostaglandins and cytokines are released from damaged fibers. Mast cells degranulate. Neutrophils arrive in the muscle within hours, followed by macrophages over the following days. These inflammatory cells release additional cytokines and chemokines, and the accumulation of inflammatory mediators sensitizes the nociceptors, the pain-sensing free nerve endings, in the muscle and its connective tissue, lowering their threshold and producing the tenderness to palpation and the ache of movement that characterizes DOMS. The inflammatory response is not purely damaging. It is the beginning of the repair process that ultimately produces stronger, more resilient muscle. The soreness is, in a sense, the cost of improvement. The muscle is paying for what it is becoming. The diaphragm is innervated exclusively by the phrenic nerve, which arises from the third, fourth, and fifth cervical nerve roots in the neck, and the clinical importance of this long, vulnerable nerve is demonstrated by the fact that spinal cord injuries at or above the level of C4 paralyze the diaphragm and require permanent ventilatory support. The phrenic nerve's origin in the neck, far above the thoracic cavity that contains the muscle it serves, is a consequence of the diaphragm's developmental history. The diaphragm develops from several mesenchymal sources, including the septum transversum, which forms in the neck region of the embryo and descends into the thoracic position during the caudal migration of the developing heart and the differential growth of the thorax. The phrenic nerve, already committed to innervating this structure, is dragged downward as the diaphragm descends from its origin near the third to fifth cervical somites to its final thoracic position, resulting in a nerve that takes an extraordinarily long intrathoracic course, entering the thorax through the superior thoracic aperture, passing lateral to the heart within the pericardium, and descending to reach the diaphragm from above. This anatomy is surgically important. The phrenic nerve can be damaged during cardiac surgery, mediastinal operations, and procedures in the neck or thorax, and is clinically important because cervical spinal cord injury at or above the level of C4 destroys the motor neuron pool from which the phrenic nerve arises, paralyzing the diaphragm and the intercostal muscles simultaneously and making spontaneous breathing impossible. The level of C4 is sometimes described as the level above which everything changes. Below it, the phrenic nerve is intact, and respiration survives even with complete motor paralysis of the limbs. Above it, the machine that keeps a person alive from breath to breath must be replaced by an external one. The single nerve that travels this long, anatomically improbable course from the neck to the muscle at the center of the chest is, in this sense, one of the most vital structures in the body. The Golgi tendon organ, a stretch receptor located at the musculotendinous junction, embedded in the collagen fibrils of the tendon, senses the tension in the tendon rather than the length of the muscle, and its inhibitory reflex action on the muscle was originally thought to be a protective mechanism against excessive force, but is now understood to have more nuanced roles in motor control. The Golgi tendon organ was classically described as a protective organ, one whose activation by high tendon tension would trigger the inverse myotatic reflex, inhibiting the muscle via IB afferent fibers and their inhibitory interneurons in the spinal cord, preventing the muscle from generating forces that could rupture the tendon. This protective role is real, and in extreme situations the Golgi tendon organ does serve to limit maximum force production. But the classical picture is now understood to be too simple. Golgi tendon organs are activated not only by the extreme forces that threaten injury but by the ordinary forces of everyday movement. They are sensitive enough to respond to the contraction of even a single motor unit, and they are continuously active during normal muscle activity, providing the central nervous system with a continuous and accurately calibrated signal of the tension in each muscle. This tension signal is distinct from the length signal provided by the muscle spindle, and together they give the nervous system a more complete picture of the mechanical state of each muscle than either could provide alone. Length and tension are the two variables that define the muscle's mechanical state, and the muscle spindle and the Golgi tendon organ are the sensory organs that measure them. In motor control, the Golgi tendon organ's IB afferents contribute to the coordination of agonist and antagonist muscles. The inhibitory signals they send to the agonist's motor neurons are simultaneously providing disinhibitory signals via the inhibitory interneuron to the antagonist muscle, helping to organize the reciprocal activation patterns that make smooth, coordinated movement possible. The Golgi tendon organ is not merely a brake. It is part of the motor control system's real-time computation of how the body is moving. Actin, the thin filament protein of the sarcomere, is also one of the most abundant proteins in virtually every non-muscle cell in the body, where it forms the dynamic cytoskeletal network that supports cell shape, enables cell migration, and mediates the division of cells during mitosis, making it one of evolution's most versatile molecular tools. The actin that polymerizes into the thin filaments of the sarcomere is, in its structure, essentially identical to the actin that forms the cytoskeletal networks of non-muscle cells, the same highly conserved protein, encoded by closely related genes, performing structurally similar roles in very different biological contexts. This conservatism reflects the fact that actin's basic properties, its ability to polymerize into filaments, to interact with myosin motor proteins, and to respond to regulatory signals, are so fundamental to eukaryotic cell biology that the protein has been maintained across hundreds of millions of years of evolution with minimal structural change. In non-muscle cells, actin filaments are organized into different structures depending on the cell type and function. The stress fibers of adherent cells that transmit force between the cytoskeleton and the extracellular matrix, the lamellipodia and filopodia of migrating cells that explore and grip the surrounding substrate, the contractile ring that divides the cell during cytokinesis, the dense cortical layer beneath the plasma membrane that maintains cell shape and resists mechanical distortion. All of these non-muscle actin structures use non-muscle myosin isoforms as their motor proteins, and the regulation of non-muscle myosin through Rho-GT-PASE signaling and myosin light chain phosphorylation is mechanistically related to smooth muscle contraction. The distinction between muscle and non-muscle is, in the language of molecular biology, less absolute than it might seem. Contraction, in some form, is a property of virtually every eukaryotic cell. The spectacular, specialized, massively amplified contraction of skeletal muscle is built on a molecular toolkit that the cell has been using, in quieter ways, for as long as there have been cells. The relationship between the muscle and its tendon is not simply an attachment but a functionally integrated mechanical system in which the stiffness of both the tendon and the muscle can be independently regulated, allowing the body to tune the overall stiffness of the musculotendinous unit for different tasks and conditions. The stiffness of a tendon, its resistance to deformation under load, is a relatively fixed material property of the collagen it is composed of, modified modestly by training and by the long-term loading history of the tendon. The stiffness of the muscle, on the other hand, can be varied rapidly and continuously by the nervous system through changes in the level of muscle activation. A fully relaxed muscle is very compliant. It offers little resistance to stretch. A maximally activated muscle is much stiffer, because the attached cross bridges and the passive elements of the sarcomere resist deformation. The ability to vary muscle stiffness independently of tendon stiffness allows the nervous system to tune the overall stiffness of the musculotendinous unit, the combined spring that connects bone to bone through the muscle-tendon system, across a wide range. During activities that require high-impact absorption, landing from a jump, running on hard surfaces, the muscles can be activated to a level that provides appropriate stiffness for the task without generating excessive force. During activities that require precise force control, threading a needle, playing a musical instrument, the muscles can be maintained at a level of activation that keeps the musculotendinous unit at the appropriate stiffness for the task. This active regulation of stiffness is one of the key mechanisms by which the musculoskeletal system achieves stability and control that passive ligamentous and bony structures alone could not provide. The stiffness of your musculoskeletal system right now, lying here in the quiet, muscles relatively relaxed, is near its minimum. The system is as compliant as it ever is. Let it be that way. Let the muscles release their remaining activation. Let the tendons bear only the weight that gravity requires of them, which tonight, in this position, is very little. You have been with me for a while now, moving through the remarkable machinery of the muscular system, the molecular motors and the calcium switches and the elastic springs that have been carrying you through every day of your life. I find that quietly wonderful. If these nights together mean something to you, if there is something in the pace of this, in the way the science becomes a kind of company for the later hours. Then subscribing to Relax with Science is a warm and simple way to say, yes, I would like more of this. I would like to come back. And I will always be here, whenever you do. The muscles of mastication, the masseter, the temporalis, the medial and lateral pterygoids, can generate bite forces of up to 600 newtons in the molar region, making the jaw one of the most powerful force-generating structures relative to its size in the body, and the masseter one of the most powerful muscles per unit cross-sectional area. The masseter is frequently cited in lists of anatomical extremes as the strongest muscle in the body when measured by the force it can exert relative to its weight. This claim requires some qualification. The maximum force a muscle can exert depends on its physiological cross-sectional area, not its weight, and many muscles produce force sufficiently without being unusually strong per unit weight. But the masseter is genuinely remarkable in the forces it generates relative to its size. It originates from the zygomatic arch in the anterior aspect of the zygomatic bone and inserts on the lateral surface of the mandible and the coronoid process. It is a multipennate muscle. Its fibers are arranged in a complex, three-dimensional pennate pattern, which gives it a high physiological cross-sectional area relative to its external dimensions and allows it to generate large forces across a relatively small range of jaw motion. The bite force of 600 newtons in the molar region, approximately 60 kilograms of force, is the combined result of the masseter, the temporalis, and the medial pterygoid working together, and represents the maximum isometric force the jaw-closing muscles can generate. During sustained, habitual clenching, bruxism, forces approaching this maximum can be sustained for extended periods, with consequences for the enamel of the teeth and the temporomandibular joints that reflect the considerable stress of sustained high-force application. The jaw muscles are fed by the trigeminal nerve's motor division, one of the few cranial nerves that carries somatic motor fibers, and the masseter reflex, elicited by tapping the chin, is one of the few monosynaptic reflexes in the cranial nerve territory, analogous to the limb tendon reflexes. Creatine phosphate, stored in the muscle and capable of rapidly regenerating ATP from ADP in the absence of oxygen, provides the energy for the first 10 to 15 seconds of maximal effort, and the discovery of its role in muscle metabolism fundamentally changed the understanding of how muscles power brief, intense activity. Creatine phosphate, also called phosphocreatine, is the muscle's immediate energy buffer, the reserve that allows the muscle to sustain maximal force production beyond the few seconds that would be possible on the stored ATP alone. At rest, the muscle stores approximately 5 times as much creatine phosphate as ATP. When maximal exercise begins and ATP is consumed faster than it can be regenerated by oxidative phosphorylation or glycolysis, the creatine kinase reaction provides the bridge. Creatine phosphate donates its high-energy phosphate group to ADP, rapidly regenerating ATP. This reaction is so fast that it can occur at the rate required by maximal exercise, and it maintains the ATP concentration in the muscle at near resting levels for 10 to 15 seconds of all-out effort, protecting the muscle from the catastrophic loss of ATP that would otherwise impair cross-bridge cycling. The depletion of creatine phosphate after 10 to 15 seconds of maximal effort is one of the primary reasons that sprinting performance begins to decline after this period. The creatine phosphate stores are replenished after exercise, much faster than glycogen, typically within a few minutes of rest, through the reverse creatine kinase reaction, using ATP regenerated by oxidative phosphorylation. The discovery of this system, by Philip and Grace Eddleton in 1927, with credit also to Cyrus Fisk and Yellowbrugda Subbaro working in parallel, was a landmark in biochemistry. Understanding the phosphagen system led directly to the use of creatine supplementation as an ergogenic aid. The supplementation of the muscle's creatine stores to provide additional buffer capacity for ATP regeneration during maximal efforts, which is, as ergogenic aids go, one of the most thoroughly evidence-supported in sports nutrition. The gluteus maximus, the largest muscle in the body by volume, is disproportionately developed in humans compared to other primates, and its primary function is not walking but the stabilization of the trunk during running and the powerful hip extension that produces the propulsion of uphill locomotion, suggesting that its development was driven by the evolution of human endurance running. The gluteus maximus occupies the back of the hip and the upper part of the buttock, arising from the iliac crest, the posterior surface of the sacrum and coccyx, and the thoracolumbar fascia, and inserting partly into the iliotibial band and partly into the gluteal tuberosity of the femur. In chimpanzees, whose gluteus maximus is much smaller relative to body size than in humans, the muscle plays primarily a posture maintenance and thigh abduction role. In humans, the gluteus maximus is dramatically enlarged, but its enlargement is specifically associated with certain activities rather than with walking generally. During level walking at normal speeds, the gluteus maximus is relatively inactive. The hip extension in the stance phase of walking is accomplished primarily by the hamstrings and by the passive extension moment of gravity. During running, however, the gluteus maximus is highly active. It fires powerfully in the stance phase, extending the hip against the flexion demand of the forward trunk lean, and it is active in the swing phase to decelerate the forward swinging leg. During climbing, lifting, and squatting, activities that require powerful hip extension against gravity, the gluteus maximus is the primary mover. Daniel Lieberman and colleagues, in their influential work on the evolution of human running, have proposed that the enlargement of the gluteus maximus in the human lineage is specifically associated with the selection for endurance running capability, because running is the activity that most demands the powerful hip extension and trunk stabilization that this muscle provides. The largest muscle in your body is, in this account, a legacy of the running that shaped the human species. The tongue is composed of 8 muscles, 4 intrinsic muscles that alter the shape of the tongue and 4 extrinsic muscles that change its position in the mouth, and their coordinated activity during speech, swallowing, and feeding represents one of the most complex neuromuscular programs in the body, developed over years of practice and ultimately executed with extraordinary speed and precision. Speech is, from a motor perspective, one of the most demanding and rapidly executed movement sequences that human beings perform. The average speaker produces approximately 15 phonemes per second in fluent speech, each phoneme requiring a specific configuration of the tongue, lips, teeth, velum, and vocal cords, and each transition between phonemes requiring the precise reshaping of these structures in milliseconds. The tongue is the most important single articulator in this system. Its intrinsic muscles, the superior and inferior longitudinal muscles, the transverse muscle, and the vertical muscle, alter the tongue shape, shortening and curling it, widening and narrowing it, flattening and thickening it. The extrinsic muscles, the genioglossus, hyoglossus, styloglossus, and palatoglossus, move the entire tongue body forward and back, down and up, into and out of the oral cavity. The genioglossus is the largest of these, arising from the mental spine of the mandible and fanning out into the tongue body and downward to the hyoid, and its fan shape allows its different regions to be activated independently, protruding the anterior tongue or retracting the posterior tongue depending on which fibers contract. The motor control of the tongue is managed primarily by the hypoglossal nerve, the 12th cranial nerve, whose nucleus in the medulla oblongata receives inputs from the motor cortex, the cerebellum, and numerous brainstem nuclei involved in the coordination of speech, feeding, and swallowing. The development of the precise, fast tongue movements required for fluent speech is a process that takes years in childhood and requires the formation of the specific neural circuits, the motor programs, that encode the movement sequences of the speaker's language. What sounds, from the outside, like effortless conversation is, in the muscle physiology of the tongue, a performance of extraordinary precision replayed thousands of times per day. The smooth muscle of the blood vessel wall, the tunica media, regulates the diameter of the vessel in response to neural signals, circulating hormones, and locally produced vasoactive substances. And this moment-to-moment regulation of vascular tone is the primary mechanism by which blood pressure is controlled. Blood pressure, the pressure of blood within the arteries, is fundamentally a product of two factors: the output of the heart and the resistance of the peripheral vessels. The heart's output is regulated by its rate and the volume it ejects with each beat. The peripheral resistance is regulated by the smooth muscle and the walls of the arterioles, the small vessels, typically 20 to 100 micrometers in diameter, that form the final common pathway to the capillary beds. The arteriolar smooth muscle responds to an enormous variety of signals. Sympathetic noradrenaline causes vasoconstriction. The smooth muscle contracts. The vessel narrows. Peripheral resistance rises. Blood pressure increases. Parasympathetic acetylcholine causes vasodilation in some vascular beds via endothelium-dependent mechanisms. Circulating adrenaline from the adrenal medulla causes vasodilation in skeletal muscle and vasoconstriction in the skin and viscera, redistributing blood to the working muscles during exercise. Angiotensin II, generated by the renin-angiotensin-aldosterone system in response to low blood pressure, causes potent systemic vasoconstriction. Locally produced nitric oxide, released from endothelial cells in response to shear stress from blood flow into various locally acting substances, causes vasodilation by activating guanylyl cyclase in the smooth muscle cell, raising cyclic GMP and ultimately reducing myosin light chain kinase activity, relaxing the muscle. The smooth muscle of the vessel wall is continuously responding to all of these signals simultaneously, finding a stable level of contraction that balances them, maintaining the vessel at a diameter that is appropriate for the current metabolic demands of the tissue it supplies and the systemic requirements of blood pressure control. This is happening in every blood vessel in your body, right now, without any input from you at all. The muscle of the uterus, the myometrium, is a smooth muscle capable of generating the most powerful sustained contractions of any smooth muscle in the body during labor, driven by the coordinated action of oxytocin receptors whose density increases dramatically in the days before delivery. The myometrium is, for most of its existence, a paradigm of smooth muscle quiescence. During pregnancy, the uterus must expand to accommodate the growing fetus without the myometrium contracting and expelling it prematurely, and an elaborate molecular machinery is responsible for maintaining this uterine quiescence. Progesterone, produced initially by the corpus luteum and then by the placenta, suppresses myometrial contractility throughout most of pregnancy by maintaining high levels of cyclic AMP in the smooth muscle cells, reducing the calcium sensitivity of the contractile apparatus and minimizing gap junction formation between cells. The shift from quiescence to the coordinated, powerful contractions of labor is mediated by a dramatic change in the uterine environment in the final days and hours before delivery. Estrogen levels rise relative to progesterone. Oxytocin receptors, whose expression in the myometrium has been increasing throughout pregnancy, reach their peak density. Gap junction formation between myometrial cells increases, creating the electrical coupling that allows action potentials to spread from cell to cell as they do in cardiac muscle, producing the coordinated, whole uterus contraction of a labor contraction. When oxytocin is released from the posterior pituitary, in response to the Ferguson reflex triggered by fetal head pressure on the cervix, it binds to these numerous receptors, triggering a phospholipase C cascade that generates inositol trisphosphate, which releases calcium from the sarcoplasmic reticulum of the myometrial cells, triggering contraction. The contractions of active labor generate intrauterine pressures of 100 millimeters of mercury or more, sustained for 30 to 60 seconds, repeating every 2 to 5 minutes, a performance of sustained smooth muscle power that has no parallel elsewhere in the body. The muscles of facial expression are unique among skeletal muscles in that they attach not to bone on both sides but to the dermis of the skin on one side, allowing them to move the skin directly and produce the facial expressions that are fundamental to human emotional communication. The facial muscles, the zygomaticus major and minor, the orbicularis oculi, the corrugator supercilii, the levator labii superioris, the depressor labii inferioris, the orbicularis oris, and many others, are developmentally derived from the second pharyngeal arch and are all innervated by the facial nerve, the seventh cranial nerve. They are unusual in their direct attachment to the dermis, which means that when they contract, they do not move one bone relative to another. They move the skin. The result is the microtopography of emotional expression, the lifting of the corners of the mouth in a smile, the furrowing of the brow in concentration or distress, the widening of the eyes in surprise, the pursing of the lips in thought. These movements are both voluntary and involuntary. The voluntary smile you produce for a photograph uses the same muscles as the involuntary smile of genuine amusement, but the pattern of muscle activation is subtly different, and observers can generally distinguish between the two. Paul Ekman's research on facial action coding showed that genuine smiles, Duchenne smiles, involve not only the zygomaticus major pulling up the corners of the mouth but also the orbicularis oculi crinkling the skin around the eyes. The orbicularis oculi component is difficult to produce voluntarily and is therefore a reliable marker of genuine positive emotion. The facial nerve carries not only the motor fibers to these muscles but also sensory fibers of taste from the anterior tongue, secretory fibers to the lacrimal, submandibular, and sublingual glands, and sensory fibers from a small area of the auricle. A remarkable convergence of functions in a single nerve that reflects the second pharyngeal arch's contribution to multiple facial structures simultaneously. The speed of muscle relaxation after a contraction is limited by the rate at which calcium can be pumped back into the sarcoplasmic reticulum, a rate-limiting step controlled by the circopumps that consume ATP, and that can be a bottleneck in high-speed repetitive contractions where relaxation must be complete before the next contraction begins. Relaxation is as important as contraction for the function of fast muscles. A muscle that cannot relax quickly cannot be reactivated quickly. And for muscles like the extraocular muscles and the middle ear muscles, whose functional demands include extremely rapid, brief contractions in rapid succession, the speed of relaxation is as critical as the speed of activation. The primary mechanism of muscle relaxation is the resequestration of calcium ions into the sarcoplasmic reticulum by the circopumps. Sarco/endoplasmic reticulum calcium ATPase, ion pump proteins that span the membrane of the sarcoplasmic reticulum and use the energy of ATP hydrolysis to transport calcium from the cytoplasm into the lumen of the reticulum against its concentration gradient. The capacity of circupumping determines the rate at which cytoplasmic calcium falls following cessation of the neural stimulus, and therefore the rate at which the troponin tropomyosin switch returns to the off state and cross-bridge formation ceases. In fast-twitch muscles, circupump density is higher than in slow-twitch muscles, a reflection of the greater relaxation speed required. The circupump is also regulated by a small protein called phospholambin, which inhibits SERCA activity in its dephosphorylated state. Phosphorylation of phospholambin by protein kinase A, activated in response to beta-adrenergic stimulation, relieves this inhibition and accelerates calcium reuptake. This is part of the mechanism by which adrenaline accelerates cardiac muscle relaxation during exercise. Faster relaxation allows faster refilling of the ventricles between beats at high heart rates. The speed of the muscle pump is the speed of the calcium removal. The calcium controls everything. Muscle cramps, the sudden, painful, involuntary contractions that can persist for seconds to minutes, are caused by abnormal spontaneous activity in the motor neurons that innervate the cramping muscle, not by a failure within the muscle itself. And their triggers include fatigue, dehydration, electrolyte imbalance, and nerve compression. The experience of a muscle cramp is familiar to almost everyone, and it is striking in its intensity relative to the apparent triviality of the usual precipitating circumstances. A brief overextension of the calf in bed, a long bout of exercise in the heat, a positional pressure on a nerve. The cramp is felt as an extraordinarily tight, painful, sustained contraction that resists voluntary attempts at relaxation and that can leave the muscle sore for hours afterward. The mechanism is neural rather than muscular. Electromyographic recordings during cramps show the involuntary high-frequency firing of the motor neurons innervating the cramping muscle, driving the muscle into sustained tetanic contraction. The factors that trigger this abnormal motor neuron firing are not fully understood, but several are well established. Muscle fatigue reduces the inhibitory input to motor neurons from Golgi tendon organs, which normally suppress motor neuron firing when tendon tension is high, and increases the excitatory input from muscle spindles, shifting the balance toward hyperexcitability and spontaneous firing. Dehydration and electrolyte imbalance, particularly reduced sodium, potassium, or magnesium, alter the electrical properties of the motor neuron membrane, lowering the threshold for spontaneous firing. The stretching of the muscle, the treatment most reliably effective for acute cramp, works by activating the Golgi tendon organs, increasing their inhibitory input to the motor neurons and suppressing the spontaneous firing. The fact that stretching works through a neural mechanism, rather than through a mechanical effect on the muscle itself, is further confirmation that the cramp originates in the motor neuron rather than in the muscle fiber. The muscle is merely following the wrong orders. The cross-bridge cycle, the sequence of attachment, power stroke, detachment, and recocking of the myosin head, is not perfectly efficient, and a fraction of cross-bridge cycles produce no net force because the myosin head detaches before completing its power stroke, a phenomenon that contributes to the heat production of muscle and to the energetic cost of sustained isometric contraction. The cross-bridge cycle can be described in several discrete steps. The strongly bound rigor state in which the myosin head is attached to actin without ATP, the release from actin upon ATP binding, the hydrolysis of ATP to ADP in inorganic phosphate, which cocks the myosin head into its pre-power stroke configuration, the attachment of the cocked head to a new site on actin, the release of inorganic phosphate, triggering the power stroke, and the release of ADP, completing the power stroke and returning to the rigor state. Each complete cycle consumes 1 ATP and theoretically produces 1 power stroke of approximately 10 nanometers. But not all cycles are thermodynamically productive. If the myosin head detaches before the release of inorganic phosphate completes the power stroke, which can occur during the rapid cycling of fast fibers, the ATP is consumed without net work being done, and the energy appears entirely as heat. Similarly, during sustained isometric contraction, where the muscle generates force without shortening, cross bridges cycle, consume ATP, and generate force, but because the muscle is not shortening, no mechanical work is done and all the energy appears as heat. This is why sustained isometric contractions, despite producing no movement, consume significant metabolic energy and generate significant heat. You can feel the warmth of a sustained grip or sustained static hold in the muscle. The heat of sustained isometric exercise and the warmth of the body generally after physical activity are, in significant part, the heat of cross-bridge cycling, the energy of ATP hydrolysis appearing as thermal energy at the site of every myosin head that fired and produced, in that particular cycle, no displacement of the actin filament. Before we continue, feel your muscles right now. Not to assess them or work them or ask anything of them. Simply feel them. The muscles of your back against the surface beneath you, as heavy and as relaxed as they will be at any point today. The muscles of your jaw, which have been working since the first word you spoke this morning and which, right now, can let the teeth part slightly, the temporomandibular joint releasing. The muscles around your eyes. The tiny muscles of the face, which carry so much of the day's unexpressed tension in their habitual microcontractions. Let them go. One by one or all at once, let them be what they are right now, which is resting. Which is exactly what muscles were made to do in the hours after dark. The peristaltic contractions of the digestive tract, the coordinated waves of smooth muscle contraction and relaxation that move food from the esophagus to the rectum, are controlled by the enteric nervous system, a network of over 500 million neurons embedded in the gut wall that operates largely independently of the brain and spinal cord. The enteric nervous system, sometimes called the gut brain, or the second brain, is, by neuron count, the largest division of the autonomic nervous system and one of the most complex neural networks outside the central nervous system. Its 500 million neurons are organized into 2 major plexuses within the wall of the gut, the myenteric plexus, lying between the longitudinal and circular muscle layers and primarily controlling motor function, and the submucosal plexus, lying between the circular muscle and the mucosa and primarily regulating secretion, blood flow, and absorption. The myenteric plexus coordinates the peristaltic reflex. A local sensory neuron detects the distension of the gut wall as a food bolus arrives, activates excitatory interneurons and motor neurons in the circular muscle proximal to the bolus, causing contraction behind the bolus, and simultaneously activates inhibitory interneurons and motor neurons in the circular muscle distal to the bolus, causing relaxation ahead of it. This coordinated behind contraction, ahead relaxation propagates along the gut as a wave, propelling the bolus forward. The entire reflex can be elicited in a segment of gut completely removed from the body and maintained in a nutrient solution. It does not require input from the vagus nerve or any central nervous system connection. The gut knows how to move food by itself. This autonomy means that the digestive system can continue functioning through sleep, through states of unconsciousness, through severing of the vagal connection, as long as the gut wall and its embedded enteric nervous system are intact. Right now, while the voluntary muscular system rests, the smooth muscle of the digestive tract is cycling through its peristaltic rhythms, moving whatever remains of today's food toward its destination, entirely without your participation. The respiratory muscles, comprising the diaphragm, the intercostals, the scalenes, the sternocleidomastoids, and the abdominal muscles, must work as a precisely coordinated group to maintain the mechanical efficiency of breathing while simultaneously adapting their contribution to posture, speech, and the varying demands of different activity levels. The respiratory system demands a particular kind of muscular compromise that is unique among organ systems. The muscles that power breathing must do their primary job continuously, without rest, for a lifetime, and they must do it while simultaneously serving as postural muscles, as muscles of speech, as muscles of coughing and laughing and straining, and as contributors to the intra-abdominal pressure that is essential for spinal stabilization. The diaphragm, as the primary inspiratory muscle, descends on inspiration and contracts simultaneously with the lumbar extensors and the transversus abdominis as part of the core stabilization system, a contraction that pressurizes the abdominal cavity and stiffens the lumbar spine against the loads of movement. When breathing demands increase, during exercise, during speech, during any activity that requires elevated ventilation, the accessory muscles of inspiration are recruited, the scalenes, which elevate the first rib, the sternocleidomastoids, which elevate the sternum, the pectorals and serratus anterior, which can elevate the ribs if the arm is fixed. The abdominal muscles become the primary active expiratory muscles during vigorous activity, contracting to drive the diaphragm upward and force air out faster than passive elastic recoil can accomplish. The coordination of all these muscles, at rest, in exercise, in speech, in posture, is achieved through a respiratory control network in the brainstem that integrates chemical information about blood gases with mechanical information from the lung stretch receptors and the respiratory muscle mechanoreceptors, with volitional override from the cerebral cortex, and with the demands of the motor systems responsible for posture and movement. The respiratory muscles, more than any others in the body, are never off duty and never doing just one thing at a time. The concept of muscle memory, the increased ease and precision with which previously learned motor skills can be performed after a period without practice, reflects genuine neural changes in the motor cortex and cerebellum that preserve the neural representations of learned motor programs even across months or years of disuse. Motor memory is not stored in the muscles. The muscles do not change in a way that encodes the movement patterns learned through practice. They are the effectors, not the storage medium. The memory is in the brain, and specifically in the changes to synaptic connectivity and cortical representation that practice produces. In the motor cortex, repeated practice of a specific movement leads to the expansion of the cortical territory devoted to the muscles involved in that movement, a well-established form of cortical plasticity demonstrated in musicians, whose hand representations in the motor cortex are enlarged relative to non-musicians, and in studies using transcranial magnetic stimulation to map motor cortical representations before and after training. In the cerebellum, practice leads to the refinement of the internal models that the cerebellum uses to predict the sensory consequences of motor commands and to generate corrective signals. The calibration of these models through practice is the cellular basis of cerebellar motor learning. At the synaptic level, long-term potentiation and depression in the corticospinal and cerebellar circuits preserve the specific pattern of synaptic weights that encodes the learned movement. These neural changes are durable. They are not easily disrupted by time away from practice, which is why a skill learned thoroughly in childhood or young adulthood can be reactivated remarkably rapidly after years of disuse. The violin player who has not played in 20 years does not start from zero. The motor cortex and cerebellum have held the movement programs in their altered synaptic weights all along, waiting. The muscles, when asked again to perform these movements, will rediscover a fluency that the neural substrate never entirely lost. The heart produces a small but measurable electrical field that can be detected at the surface of the body by electrodes placed on the skin, the electrocardiogram. Because the synchronized electrical activity of the 250 million cardiomyocytes of the ventricles generates a dipole field that propagates through the conductive tissues of the body to the skin surface. The electrocardiogram is one of medicine's most powerful diagnostic tools, and it works because the heart is a large, coordinated electrical source surrounded by the electrically conductive medium of the body's tissues. When the sinoatrial node fires and the wave of depolarization spreads across the atria, recorded as the P wave, and then, after the atrioventricular node's delay, propagates through the bundle of His, the bundle branches, and the Purkinje fibers to activate the ventricular myocardium, recorded as the QRS complex, and when the ventricles then repolarize, recorded as the T wave. Each of these events generates a brief electrical dipole in the body, whose orientation and magnitude can be sensed by electrodes on the skin. The 12-lead ECG, with electrodes in standardized positions on the limbs and chest, samples this dipole field from 12 different directions, providing a three-dimensional picture of the electrical activity of the heart. The shape, duration, and amplitude of each wave in the ECG encodes information about the anatomy and the electrical function of specific regions of the myocardium. The ST segment elevation of myocardial infarction reflects the altered resting membrane potential of ischemic muscle. The broad QRS complex of bundle branch block reflects the abnormal, slow spread of activation through the ventricular muscle when the rapid conduction system is disrupted. The prolonged QT interval of certain channelopathies reflects delayed ventricular repolarization that predisposes to arrhythmia. All of this information, the electrical biography of the beating heart, is readable in the waveforms generated by the coordinated electrical activity of millions of cardiomyocytes, detectable at the skin surface by the same physics that makes any electrical dipole generate a field that propagates through space. The heart is always broadcasting. The skin is always receiving. The maximum voluntary contraction of a muscle is not determined solely by the number and size of its motor units, but also by the degree of voluntary activation, the percentage of available motor units that the nervous system can actually recruit under maximal effort, which in untrained individuals is typically 80 to 95%, leaving a reserve that training can increase. The force a muscle can voluntarily produce is, in theory, limited by the force of all its motor units firing maximally together. In practice, the voluntary force falls short of this theoretical maximum because the nervous system does not recruit all available motor units even during maximal effort. This gap between actual voluntary force and the theoretical maximum is quantified as voluntary activation, the percentage of maximal motor unit activity that voluntary effort can achieve. In untrained individuals, this is typically in the range of 80 to 95%, meaning that 5 to 20% of the muscle's force capacity is effectively inaccessible to voluntary command. Training, particularly strength training and practices that require maximal effort, sprint training, powerlifting, certain forms of neural activation training, can increase voluntary activation toward 100%. Allowing more of the muscle's theoretical capacity to be expressed in actual force production. This is why the early gains in strength from resistance training are largely neural rather than structural. The muscle does not hypertrophy appreciably in the first weeks of training, but strength increases substantially because the nervous system learns to recruit more motor units and to synchronize their firing more effectively. There is also evidence from electrical stimulation studies that superimposing an electrical stimulus on a maximal voluntary contraction can still produce an additional force increment, the twitch superimposition technique, and that this additional force, which the voluntary system could not produce alone, is reduced by training. The nervous system learns to approach its own limits more closely. The muscle has, all along, been capable of more than the nervous system was allowing it to do. The stretch shortening cycle, the sequence of eccentric loading followed by concentric contraction, produces greater force and greater power output than a concentric contraction from rest, because of both the elastic energy storage in the tendon and the potentiation of contractile force through the stretch reflex and tighten-based mechanisms. The stretch shortening cycle is the fundamental movement pattern of virtually all powerful human actions: running, jumping, throwing, catching. The pattern is pre-activation of the muscle before ground contact or impact. Eccentric loading as the contact force stretches the muscle tendon unit. Then concentric contraction as the body responds and drives the movement forward. The advantage of this sequence over a pure concentric contraction from rest is substantial, and it comes from multiple mechanisms acting simultaneously. The elastic energy stored in the tendon during the eccentric loading phase is returned during the subsequent concentric phase, supplementing the contractile energy and reducing the metabolic demand. The stretch reflex, triggered by the rapid lengthening that activates muscle spindles, provides additional motor neuron drive during the concentric phase, augmenting the voluntary activation. Tighten, actively stiffened by calcium released in the pre-activation phase, stores additional elastic energy during the eccentric stretch and returns it during concentric shortening. The net result is that the power output of a jump preceded by a counter movement, a standard squat jump, is approximately 10 to 20% greater than a jump from a static crouching position, even though the initial muscle lengths are similar. Sprinters, basketball players, gymnasts, all of them exploit the stretch shortening cycle in essentially every explosive movement they make, the muscle tendon unit acting as a spring that is loaded and released in the service of power production. The body does not generate its most powerful movements from a standstill. It loads first. It prepares. It stores. And then it releases. The soleus muscle, the deep calf muscle that lies beneath the gastrocnemius, is considered by some physiologists to be more important for sustained standing and walking than the larger, more prominent gastrocnemius, because it is composed almost entirely of slow-twitch fibers and can sustain the postural support of the ankle joint throughout the hours of upright activity without fatiguing. The soleus is invisible in the way that the most essential things often are. It lies beneath the gastrocnemius, the prominent calf muscle whose twin heads are visible as the rounded prominence of the calf when the muscle is contracted, and it is largely hidden from view. It arises from the posterior surface of the upper tibia and fibula, lower and deeper than the gastrocnemius's femoral origins, and shares the Achilles tendon with the gastrocnemius, inserting onto the calcaneal tuberosity. Because the soleus does not cross the knee joint while the gastrocnemius does, their contributions to plantar flexion vary with knee angle. With the knee flexed, the gastrocnemius is relatively ineffective and the soleus provides the majority of plantar flexion force. In standing, the ankle joint must generate a continuous plantar flexion moment to balance the dorsiflexion moment of gravity acting through the body's center of mass, which is typically positioned anterior to the ankle joint. This continuous demand falls primarily on the soleus. The gastrocnemius, being a fast muscle, would fatigue rapidly if it were required to sustain this moment continuously. The soleus, composed predominantly of type 1 slow-twitch fibers, can sustain this low-level continuous activation for hours without significant fatigue. It is the standing muscle, the muscle that keeps you upright through the long hours of the day by maintaining the ankle against gravity's forward pull. Tonight, Lying horizontal, the gravitational demand on the soleus is near zero. For perhaps the first time in the waking hours, it is truly resting. And so, in a small way, are you? The intrafusal muscle fibers of the muscle spindle, the specialized fibers that contain the sensory endings of the IA and two afferents, are not vestigial remnants of the regular contractile machinery but active structures whose gamma motor neuron innervation allows the sensitivity of the spindle to be adjusted continuously to match the mechanical demands of the current task. The gamma motor neurons, sometimes called the fusomotor system, because they innervate the fusomotor or intrafusal fibers of the muscle spindle, are small diameter motor neurons in the ventral horn that receive descending inputs from the motor cortex, the cerebellum, and various brainstem nuclei but do not contribute to the force-generating capacity of the extrafusal muscle. Their sole function is to regulate the sensitivity of the muscle spindle by adjusting the tension of the intrafusal fibers. There are two types of gamma motor neurons: dynamic gamma neurons, which innervate BAG1 intrafusal fibers and increase the sensitivity of the spindle to the rate of change of length, enhancing the spindle's dynamic sensitivity, and static gamma neurons, which innervate chain fibers and some BAG2 fibers and maintain the spindle's sensitivity to steady-state length. This independent control of spindle sensitivity allows the fusomotor system to preset the spindle for the expected conditions of each task, increasing dynamic sensitivity before movements that require rapid reflex responses to unexpected perturbations, increasing static sensitivity during tasks that require sustained position control. The activity of the gamma motor neurons is coordinated with the activity of the alpha motor neurons that drive the extrafusal muscle. The alpha-gamma coactivation that occurs during voluntary movement ensures that the spindles remain sensitive throughout the movement, even as the muscle shortens and the spindle would otherwise go slack. The intrafusal fibers, contracting under gamma drive, take up the slack in the spindle as the extrafusal fibers shorten, maintaining the spindle at its optimal sensitivity throughout the range of motion. The spindle is not a passive sensor. It is an actively regulated instrument. The cardiac output of the heart at rest, approximately 5 liters per minute, can increase to 25 liters per minute or more during maximal exercise in trained athletes, representing a 5-fold increase achieved through simultaneous increases in both heart rate and stroke volume, and reflecting the cardiac muscle's extraordinary capacity for adaptation. 5 liters per minute is the amount of blood your heart is moving right now, approximately. Every minute, the entire blood volume of the body circulating once through the cardiovascular system, delivered to the lungs for oxygenation and then distributed to the tissues for metabolism. This is already a remarkable performance. 5 liters per minute, maintained without interruption, day and night, for the duration of a lifetime. But the cardiac muscle's capacity extends far beyond the resting output. In trained endurance athletes at maximal exercise, cardiac output can exceed 35 liters per minute, a 7-fold increase over rest. This increase is achieved through two mechanisms: an increase in heart rate, from the resting 60 beats per minute to the maximal 200 or more, driven by sympathetic activation and the withdrawal of vagal tone, and an increase in stroke volume. The amount of blood ejected with each beat, from the resting 70 milliliters to perhaps 120 milliliters or more in trained athletes. The increase in stroke volume comes from increased filling of the ventricles during the shortened diastole, achieved through the increased venous return of exercise and the more complete emptying of the ventricles with each beat due to increased contractility. The cardiac muscle itself changes with endurance training. The ventricles enlarge, accommodating a greater end diastolic volume, and the muscle becomes more efficient at generating the force required to eject the larger stroke volume. The resting heart rate of elite endurance athletes can fall below 40 beats per minute, reflecting the cardiac adaptation that allows the heart to pump adequate flow at rest with fewer, more powerful beats. The cardiac muscle is, in its capacity for adaptation, the most plastic muscle in the body, continually remodeling its architecture in response to the demands placed on it, becoming more capable with each week and each month of sustained training. The muscles responsible for maintaining upright posture, the anti-gravity muscles, include the soleus, the erector spinae, the hip extensors, and many smaller deep muscles, and they are characteristically composed of a high proportion of slow-twitch fibers, a structural adaptation that allows the continuous low-level activation of postural maintenance without metabolic exhaustion. Posture is, in muscular terms, a performance that never ends. From the moment you rise in the morning to the moment you lie down at night, the anti-gravity muscles are in continuous low-level contraction, resisting the tendency of the body to collapse toward the floor under the pull of gravity. The loads they bear are modest compared to the loads of vigorous exercise, the soleus resisting the dorsiflexion moment at the ankle, the erector spinae resisting the flexion moment at the lumbar spine, the gluteus medius preventing the pelvis from dropping on the unsupported side during walking. But the duration of this activity means that fatigue resistance is the critical property, more important than maximal force capacity. The slow-twitch fiber composition of the postural muscles reflects this requirement precisely. Type 1 fibers generate their ATP oxidatively, at high efficiency, and can sustain their contraction for hours on a continuous supply of oxygen and fat, the body's most energy-dense fuel. They contain abundant mitochondria and myoglobin, and they are well supplied with capillaries. They do not accumulate lactic acid or inorganic phosphate at the rates that fast-twitch fibers do during sustained activity, because their cross-bridge cycling rate is lower and their metabolic demands are met efficiently by oxidative phosphorylation. The anti-gravity muscles are, in this metabolic sense, the exact opposite of the sprinting muscles: slower, quieter, more efficient, built for duration rather than peak performance. Tonight, with the body horizontal, they are released from their postural duty for the first time since morning. The erector spinae that has been maintaining the lumbar curve through every standing and sitting moment of the day is finally unloaded. Let it rest completely. It has been working since you opened your eyes. The tongue muscle of the genioglossus is one of the most important muscles in the context of sleep-disordered breathing. Its tendency to relax during sleep can allow the posterior tongue to fall back against the posterior pharyngeal wall, narrowing or occluding the airway and producing the snoring and apnea of obstructive sleep apnea. During waking, the genioglossus maintains a tonic level of contraction that holds the tongue forward in the oral cavity, keeping the base of the tongue away from the posterior pharyngeal wall and maintaining the patency of the upper airway. This tonic activity is driven by direct inputs from the respiratory centers of the brainstem to the hypoglossal nucleus, and it increases in proportion to the respiratory drive. During periods of elevated CO2 or reduced O2, genioglossus activity increases, holding the airway open against the negative pressure generated during inspiration. During sleep, however, and particularly during the rapid eye movement stage of sleep, this tonic activity falls dramatically. In people with a narrow upper airway, from obesity, from structural factors, from the normal aging-related changes in upper airway anatomy, the fall in genioglossus tone during sleep can be sufficient to allow the tongue and soft palate to obstruct the airway partially or completely with each breath. The partial obstruction produces snoring, the vibration of the soft tissues in the narrowed airway under the pressure gradient of inspiration. Complete obstruction produces an apnea, a cessation of airflow, typically lasting 10 to 60 seconds, terminated when the rising CO2 level arouses the sleeper enough to restore muscle tone and reopen the airway. Obstructive sleep apnea affects a substantial proportion of the adult population and has significant cardiovascular and metabolic consequences from the recurrent oxygen desaturation and sleep fragmentation it produces. One muscle, one pair of genioglossi, whose relaxation during sleep is the proximate cause of a condition affecting millions of people worldwide. The muscle does not know it is doing harm. It is simply resting, as muscles do at night. The problem is purely architectural. The airway into which it is resting is not wide enough to accommodate the relaxation. The smooth muscle of the iris, comprising the sphincter pupillae and the dilator pupillae, controls the diameter of the pupil in response to light intensity, regulating the amount of light entering the eye, and the speed and completeness of the pupillary light reflex is one of the most diagnostically useful signs in clinical medicine. The pupil is the aperture of the eye, and its diameter, which ranges from approximately 2 millimeters in bright light to 8 millimeters in darkness, is determined by the balance of two smooth muscles with opposing actions. The sphincter pupillae is a ring of circular smooth muscle around the pupillary margin, innervated by parasympathetic fibers from the ciliary ganglion via the oculomotor nerve, whose contraction reduces the pupil diameter, miosis. The dilator pupillae is a sheet of radially oriented smooth muscle, innervated by sympathetic fibers from the superior cervical ganglion, whose contraction increases the pupil diameter, mydriasis. The pupillary light reflex, the constriction of the pupil in response to illumination of the retina, is mediated by a pathway that runs from the retina through the optic nerve to the pretectal nucleus in the midbrain, which drives bilateral Edinger-Westphal nuclei, which innervate both ciliary ganglia via the oculomotor nerve, producing bilateral constriction. Both the illuminated eye and the fellow eye constrict equally, the direct and consensual responses respectively. The clinician uses this reflex in several important ways. In a comatose patient, absent or asymmetric pupillary responses provide information about the level and laterality of brainstem compression. A dilated, unreactive pupil on one side in a patient with falling consciousness suggests uncal herniation. Compression of the ipsilateral oculomotor nerve by the uncus of the temporal lobe being forced through the tentorium cerebelli by rising intracranial pressure. The RAPD, relative afferent pupillary defect, detected by the swinging flashlight test, indicates an asymmetry in the afferent limb of the reflex, indicating optic nerve or retinal disease on one side. The two small smooth muscles of the iris are, in these clinical situations, windows into the state of the brainstem. The muscles of the rotator cuff, the subscapularis, supraspinatus, infraspinatus, and teres minor, do not function primarily as prime movers of the shoulder but as dynamic stabilizers of the glenohumeral joint, compressing the humeral head into the glenoid fossa with every shoulder movement to prevent the instability that the joint's shallow socket would otherwise permit. The rotator cuff is not a single muscle but a group of 4 muscles whose tendons merge into a broad, continuous reinforcement of the joint capsule of the shoulder, the rotator cuff tendon. The subscapularis covers the anterior surface of the scapula and inserts on the lesser tuberosity of the humerus, acting as an internal rotator and anterior stabilizer. The supraspinatus occupies the supraspinous fossa above the spine of the scapula and inserts on the superior facet of the greater tuberosity, initiating abduction and compressing the superior aspect of the joint. The infraspinatus occupies the infraspinous fossa below the spine of the scapula and inserts on the middle facet of the greater tuberosity, externally rotating and stabilizing the posterior aspect of the joint. The teres minor, the smallest of the 4, lies below the infraspinatus and inserts on the inferior facet of the greater tuberosity, also externally rotating and contributing to posterior stabilization. Together, the rotator cuff muscles form a muscular cuff whose coordinated contraction during shoulder movement provides a compressive force that maintains the humeral head in contact with the glenoid fossa against the destabilizing forces generated by the larger prime movers, the deltoid, the pectorals, the latissimus. Without the rotator cuff compression, the superior pull of the deltoid during arm elevation would drive the humeral head superiorly, impinging the rotator cuff tendons against the acromion above and precluding effective elevation. With an intact rotator cuff, the compressive and inferior directed force of the cuff muscles balances the superior force of the deltoid, allowing the deltoid's abductive torque to elevate the arm smoothly. Rotator cuff tears, which affect a substantial proportion of adults over 50, disrupt this balance, producing the characteristic painful arc and weakness of rotator cuff pathology. The mechanism by which the muscle fibers plasma membrane, the The sarcolemma is mechanically coupled to the extracellular matrix is through a protein complex called the dystrophin-associated protein complex. In the genetic absence of dystrophin, the principal structural protein of this complex, is the cause of Duchenne muscular dystrophy, the most common muscular dystrophy. The sarcolemma of the muscle fiber is subject to enormous mechanical stress during contraction. The forces generated inside the cell must be transmitted outward to the extracellular matrix and the connective tissue framework without tearing the membrane that separates the cellular contents from the extracellular environment. The dystrophin-associated protein complex is the bridge that accomplishes this transmission. Dystrophin itself is a large, rod-shaped protein at approximately 3,400 amino acids, one of the largest proteins in the body, that links the intracellular actin cytoskeleton to a complex of glycoproteins in the sarcolemma, which in turn link to the extracellular matrix protein laminin. This mechanical linkage distributes the contractile forces laterally, protecting the sarcolemma from the stress concentrations that would otherwise occur at the points where the force is transmitted from the contractile apparatus to the tendon. Without dystrophin, as in Duchenne muscular dystrophy, this mechanical link is absent. Each contraction creates stress concentrations in the sarcolemma that produce microinjuries, small membrane tears that allow calcium to enter the fiber from the extracellular space, activating proteases, triggering inflammation, and eventually killing the fiber. The satellite cells regenerate new fibers initially, but the continuous cycle of injury and regeneration eventually exhausts the satellite cell population, and the muscle tissue is progressively replaced by fibrotic connective tissue and fat. The clinical consequence: progressive muscle weakness beginning in early childhood, typically affecting the proximal leg muscles first, progressing to the arms and trunk, and eventually the respiratory muscles, reflects this progressive replacement of functional muscle by non-contractile tissue. Duchenne muscular dystrophy demonstrates, with tragic clarity, how essential the mechanical integrity of the sarcolemma is to the survival of the muscle fiber. The muscle's blood supply is not constant during exercise but is dynamically regulated through a process called functional hyperemia, the increase in blood flow to an active muscle that precisely matches the increased metabolic demand, through a combination of local, neural, and hormonal mechanisms that together achieve a remarkably close coupling between oxygen delivery and oxygen consumption. At rest, the skeletal muscle receives approximately 15 to 20% of the cardiac output, or about 1 liter per minute. During maximal exercise, this rises to 80 to 85% of the dramatically increased cardiac output, up to 25 liters per minute in trained athletes, representing a blood flow to skeletal muscle of 15 to 20 liters per minute, an increase of 15 to 20-fold. This redistribution is achieved through the combination of the generalized increase in cardiac output and the specific vasodilation of the resistance vessels in the active muscles, which reduces their vascular resistance and allows them to capture a much larger fraction of the available flow. The vasodilation of the active muscle vasculature is triggered by multiple locally produced vasodilators. the fall in local oxygen tension as the active muscle extracts oxygen faster than it is delivered, the rise in CO2 and H+ from increased metabolism, the release of potassium from the repeatedly depolarizing muscle fibers, the local production of adenosine, a potent vasodilator released when ATP is consumed faster than it can be regenerated, the release of nitric oxide from the endothelium in response to the increased shear stress of the rising blood flow. Each of these signals reflects a different aspect of the muscle's increased metabolic activity, and each drives vasodilation by a slightly different mechanism, providing a redundant and robust system for matching delivery to demand. The result is that even at very high exercise intensities, the working muscle's oxidative capacity is rarely limited by blood flow. The vasodilatory system is sufficiently responsive and sufficiently powerful to maintain oxygen delivery. The bottleneck, when it exists, is usually the heart's cardiac output or the mitochondria's capacity for oxidative phosphorylation, not the supply of blood to the vessel. The evolution of the human vocal tract, including the lowering of the larynx, the lengthening of the pharynx, and the development of the precise neural control of the laryngeal muscles, required changes not just in the bony structures but in the muscular and neuromuscular systems that produce the range of sounds required for language. The human larynx is positioned lower in the throat than in other primates, a descent that completed over the course of human evolution and continues during the first years of each human life, as the infantile larynx descends from the high position at birth, which allows infants to breathe and nurse simultaneously, to the adult position by the age of 2 to 3. This lowered larynx position creates the long, curved pharynx that acts as a resonant cavity whose shape can be modified by the tongue, the lips, the velum, and the laryngeal muscles to produce the rich variety of formants, frequency resonances, that distinguish the vowel sounds of human speech. The muscles of the larynx, the intrinsic laryngeal muscles, innervated by the recurrent laryngeal and superior laryngeal branches of the vagus nerve, control the length, tension, and position of the vocal folds with extraordinary precision, modulating the fundamental frequency of the voice and determining the quality of phonation. The cricoarytenoid muscles abduct and adduct the vocal folds, opening and closing the glottis. The cricothyroid muscle lengthens and tenses the vocal folds, raising their fundamental frequency for higher pitches. The thyroarytenoid muscle shortens and thickens the vocal folds. The remarkable precision of these muscles, capable of adjusting vocal fold tension to the degree required to produce pitches separated by fractions of a semitone, and to transition between phonatory states in milliseconds, is the product of both the high density of muscle spindles in the laryngeal muscles and the fine-grained cortical motor representation devoted to their control. The voice is one of the human body's most elaborate muscular achievements, and the muscles that produce it are among the most precisely controlled in the body. The phenomenon of rigor mortis, the stiffening of muscles that occurs after death, is caused by the permanent formation of actin-myosin crossbridges in the absence of ATP, and its onset, progression, and resolution follow a predictable time course that has been used by forensic pathologists to estimate time of death since the 19th century. Rigor mortis is the endpoint of the cross-bridge cycle, the state the sarcomere reaches when ATP is depleted and the mechanism for cross-bridge detachment no longer operates. In the living muscle, the normal resting state is one in which most cross-bridges are either in the detached state or cycling through their normal sequence. ATP binding to the myosin head is what drives detachment from actin. Without ATP, the myosin head cannot release the actin filament, and the actin and myosin remain locked in the strongly bound rigor state. This is why the rigor state is so named. It is the state of maximum actin-myosin bonding, maximum cross-bridge stiffness, and maximum resistance to passive deformation. After death, as the circulation ceases and aerobic metabolism fails, the muscle's ATP stores are consumed, first supplemented by creatine phosphate, then briefly by anaerobic glycolysis, until ATP is depleted, typically within 6 to 12 hours depending on temperature and the state of the muscle at death. As ATP falls, cross bridges cannot be released, the actin-myosin bonds accumulate, and the muscle stiffens. The stiffening typically begins in the smaller muscles of the face and neck and progresses to the larger muscles of the trunk and limbs over the following hours. The resolution of rigor mortis, typically 24 to 48 hours after onset, is not caused by ATP recovery. It is caused by the autolytic degradation of the muscle proteins themselves, as the enzymes released from lysosomes begin to break down the actin and myosin filaments and disrupt the cross bridges physically. The muscle softens as it decomposes. Rigor mortis, from beginning to end, is the last act of the muscle's molecular machinery. The muscles of the pelvic floor and the external urethral sphincter must maintain their tonic contraction to prevent urinary leakage during the increases in intra-abdominal pressure that accompany coughing, sneezing, laughing, and lifting. And the reflex that coordinates this continuous guarding requires the integration of sensory information from the bladder and urethra with motor output to the skeletal muscles of the perineum. Continence, the ability to retain urine and feces until the appropriate time and place, requires the continuous, coordinated activity of both smooth muscle and skeletal muscle components in a system that operates largely below conscious awareness but can be overridden by voluntary control when required. The bladder's detrusor muscle, smooth muscle, must remain relaxed and compliant as the bladder fills, accommodating increasing volumes at low pressure. The internal urethral sphincter, also smooth muscle, under sympathetic control, provides resting closure. The external urethral sphincter, skeletal muscle, under pudendal nerve control, provides additional voluntary closure and is the muscle responsible for the volitional interruption of urination. The pelvic floor muscles, particularly the pubococcygeus and puborectalis, wrap around the urethra and provide a kinking mechanism that adds to urethral resistance when they contract. Coughing, sneezing, laughing, and lifting all produce sudden, large increases in intra-abdominal pressure that are transmitted equally to the bladder and to the urethra. If the urethral closure mechanism does not respond to these increases instantaneously, the pressure differential between bladder and urethra would drive urine outward. The reflex that prevents this is a preemptive contraction of the external urethral sphincter and pelvic floor that is triggered by the signals that precede the cough, the initial inspiration, the glottal closure, and that is complete before the peak abdominal pressure arrives. This anticipatory contraction is learned. It develops gradually in childhood as the child gains continence, and it relies on the integrity of the pudendal nerve and the spinal continence center in the sacral cord. Its failure in the context of pelvic floor muscle weakness or neurological deficit is the mechanism of stress urinary incontinence, one of the most common conditions affecting quality of life in adults worldwide. The speed of contraction of a single muscle fiber can be characterized by its maximum shortening velocity, the velocity at which the sarcomeres shorten when no external load is applied, and this parameter varies by approximately tenfold between the slowest and fastest muscle fibers in the human body, from the soleus slow fibers to the extraocular fast fibers. The tenfold range in maximum shortening velocity across fiber types reflects the tenfold range in myosin ATPase rate, which determines how rapidly each power stroke cycle can be completed. In the slow twitch soleus fibers, expressing MHC beta slow, the ATPase rate is approximately 1 power stroke per second per myosin head, a slow rate that produces a maximum shortening velocity of about 1 muscle fiber length per second. In the fast-twitch extraocular fibers, expressing MHC-IIB in non-human species, or MHC-IID/I9 in humans, the ATPase rate is approximately 10 times greater, producing shortening velocities of approximately 10 fiber lengths per second. In terms of absolute shortening velocity, the extraocular muscle fibers can shorten at approximately 60 to 70 millimeters per second given their fiber lengths, fast enough to produce the 600 degree per second angular velocities of the most rapid saccades, accounting for the mechanical advantage of the extraocular muscles of the globe. The intermediate fiber types occupy the range between these extremes, and the mixture of fiber types in a given muscle determines the overall velocity of the muscle's contraction. A muscle composed predominantly of slow fibers will contract at close to the slow fiber velocity, one composed predominantly of fast fibers at close to the fast fiber velocity. The mixed fiber composition of most human limb muscles places them at intermediate velocities, appropriate for the mixed velocity demands of daily activity. The body has, in its fiber type distribution, made a different computational decision for each muscle, allocating fast fibers where speed is needed and slow fibers where endurance is needed, with the specific allocation reflecting the evolutionary and developmental history of each muscle's use. The concept of the physiological cross-sectional area of a muscle, distinct from its anatomical cross-sectional area, accounts for the pennation angle of the fibers and provides a measure that is directly proportional to the maximum force the muscle can generate, since force is generated by the cross bridges in each fiber and is therefore proportional to the total cross-sectional area of all fibers summed perpendicular to their long axes. Understanding muscle force from anatomy requires the concept of physiological cross-sectional area, because the anatomical cross-section of a pennate muscle, the cross-section perpendicular to the long axis of the whole muscle, does not capture the total area of all the muscle fibers. In a parallel fibered muscle, the anatomical and physiological cross-sections are the same, because all fibers run along the long axis of the muscle and their cross-sections contribute fully to the anatomical cross-section. In a pennate muscle, the fibers run at an angle to the long axis, and the anatomical cross-section cuts each fiber at an oblique angle, underestimating the total fiber cross-sectional area. The physiological cross-sectional area is calculated as the muscle volume divided by the fiber length, a formula that gives the total area of all fibers cut perpendicularly to their long axes, regardless of the pennation arrangement. This measure is what determines force, since each square centimeter of fiber cross-section contains approximately the same number of sarcomeres in parallel, and since each sarcomere contributes approximately the same maximum force, the maximum force of the whole muscle is proportional to its physiological cross-sectional area, multiplied by the cosine of the pennation angle to account for the component of fiber force acting along the muscle's line of pull. For typical pennation angles, this cosine factor is between 0.9 and 1.0, a relatively modest correction. The practical consequence is that the force of a pennate muscle is greater than its external size suggests. The quadriceps and the gastrocnemius generate more force than parallel fibered muscles of the same volume would, because pennation allows more fiber cross-sectional area to be packed into the same muscle volume. The architecture of the muscle is an engineering decision, and its evaluation requires the right measure of the relevant dimension. There are so many places left to go in the body's remarkable muscular world: the chemistry of energy systems, the neurology of voluntary movement, the extraordinary diversity of muscle types and roles across every organ and tissue. If you would like to be here for those journeys, and for all the other quiet wonders that relax with science brings to the night, one slow evening at a time, subscribing keeps the door open. You will find something waiting on an evening when you need it most. Made slowly. Made carefully. Made for exactly this hour. The muscle's response to disuse, the atrophy that occurs during immobilization, bed rest, or spaceflight, is the reverse of hypertrophy, involving a reduction in fiber cross-sectional area driven by a net increase in protein degradation over protein synthesis, mediated by the MAFBX and MOF1 ubiquitin ligases of the ubiquitin proteasome pathway. Muscle mass is not maintained without cost. The synthesis and maintenance of the contractile proteins, actin, myosin, titin, and all the others, requires continuous protein turnover, a process in which existing proteins are degraded and replaced with newly synthesized ones. In a muscle that is being used, the rates of synthesis and degradation are approximately balanced, or synthesis slightly exceeds degradation, maintaining mass. In an unused muscle, the balance tips toward degradation, and mass falls. The primary pathway of muscle protein degradation during disuse is the ubiquitin proteasome system, a cellular protein disposal mechanism in which proteins destined for degradation are tagged with chains of ubiquitin molecules by specific ubiquitin ligase enzymes and then fed into the proteasome, a large, barrel-shaped protease complex that unfolds and cleaves the tagged protein into small peptide fragments. The two ubiquitin ligases most important in muscle disuse atrophy, MAFBX, also called atrogen 1, and MOF1, are specifically induced in atrophying muscle and are now used as molecular markers of atrophic conditions. Their expression is suppressed by insulin-like growth factor 1 and by mechanical loading, the same signals that promote anabolic responses and hypertrophy, and induced by glucocorticoids, inflammatory cytokines, and the absence of mechanical loading. Spaceflight astronauts lose muscle mass rapidly in the first weeks of weightlessness, primarily from the posterior leg and hip muscles that normally sustain posture against gravity, with MOF1 and MAFBX expression increased dramatically. The muscle, without the mechanical signal of loading, concludes that it is carrying more mass than its work requires, and begins to reduce. It is a metabolism of precision. The body maintains exactly the muscle it uses and no more. The phenomenon of post-activation potentiation, the transient increase in twitch force following a conditioning tetanic contraction of the same muscle, is caused by the phosphorylation of the myosin regulatory light chains by calcium calmodulin-dependent myosin light chain kinase, which increases the calcium sensitivity of the cross-bridge and the rate of force development. Post-activation potentiation is one of the more counterintuitive phenomena in muscle physiology. After a muscle has been subjected to a brief, near-maximal tetanic contraction, a subsequent submaximal twitch, exactly the same stimulus as before the tetanus, produces a greater force than it did before the conditioning contraction. The muscle, which has just been working hard, is temporarily stronger in response to the same stimulus. This is not the long-term hypertrophic response to training, which takes weeks to develop. It is an acute effect, developing within seconds and lasting minutes to tens of minutes. The mechanism is the phosphorylation of the regulatory light chains of myosin. The high intracellular calcium of the tetanic contraction activates calmodulin, which activates myosin light chain kinase, which phosphorylates a specific serine residue on the myosin regulatory light chain. This phosphorylation changes the orientation of the myosin heads relative to the thick filament backbone, swinging them away from the thick filament surface and making them more available for actin binding. The calcium sensitivity of the cross-bridge is increased. The same amount of calcium released from the sarcoplasmic reticulum now produces more cross-bridge formation than before the conditioning contraction. The rate of force development is faster. The twitch force is greater. In athletic practice, this phenomenon is exploited through contrast training in complex training protocols. Heavy strength exercises paired with explosive power exercises. in which the potentiating effect of the heavy lift is used to enhance the force production of the subsequent explosive movement. The athlete who performs a heavy squat before a vertical jump may jump higher than without the preceding squat, because the squat has potentiated the quad and hip extensor muscles through this mechanism. The muscles of the eye, through their connection with the vestibular system via the vestibulo-ocular reflex, maintain gaze stability during head movements by producing compensatory eye movements that are equal and opposite to the head movements, allowing the visual world to remain stable on the retina even as the head moves. The vestibulo-ocular reflex, the VOR, is one of the fastest and most reliable reflexes in the body, with a latency of approximately 10 to 15 milliseconds, compared to the visual system's 200 millisecond processing time. Its speed is essential because the VOR must compensate for head movements in near real time, generating eye movements that prevent the visual world from blurring on the retina before the visual system could even detect that blurring was occurring. The reflex arc is trisynaptic. The semicircular canals of the vestibular apparatus detect rotational head acceleration through the displacement of endolymph. The hair cells of the ampullae transduce this displacement into afferent signals in the vestibular nerve, and these signals project to the vestibular nuclei in the brainstem. From the vestibular nuclei, the reflex pathway projects to the motor nuclei of the extraocular muscles, the oculomotor, trochlear, and abducens nuclei, through the medial longitudinal fasciculus, a fiber tract that runs up the brainstem from the vestibular nuclei to the eye muscle nuclei. The motor neurons of these nuclei drive the extraocular muscles to produce the compensatory eye movement. The gain of the VOR, the ratio of eye movement velocity to head movement velocity, is approximately 1 under normal conditions, meaning the eye moves at exactly the same velocity as the head but in the opposite direction, maintaining gaze on the same point in space. This gain is calibrated by the cerebellum, which compares the visual feedback of image stability on the retina with the vestibular signal of head movement and adjusts the VOR gain to minimize retinal image slip. The VOR has been maintaining your gaze stability through every head movement you have made today, in complete silence and complete invisibility, doing its work in the brainstem with no involvement from the conscious mind whatsoever. The muscles of the lower limbs, during walking, consume only a fraction of the metabolic energy that simple biomechanical calculations would predict, because the musculoskeletal system is organized as an inverted pendulum during stance phase, the body's center of mass vaulting over the stance leg like a pole vault, and as a pendulum during swing phase, with the swinging leg exchanging kinetic and potential energy with near-perfect efficiency. The metabolic efficiency of human walking is higher than any simple account of the muscular work done against gravity and ground reaction forces would predict. And the explanation lies in 2 energy conservation mechanisms that operate in alternation with each step. During the stance phase of walking, when one foot is on the ground and the body vaults over it, the body behaves as an inverted pendulum. The center of mass rises as it passes over the stance foot and falls as the foot strikes and leaves the ground. This rise and fall involves a continuous exchange between kinetic energy, velocity, and gravitational potential energy, height, analogous to the energy exchange in a pendulum but inverted. Energy that would have been lost to deceleration is stored as potential energy at the peak of the arc and returned as kinetic energy during the descent. The efficiency of this exchange reduces the muscular work required to maintain forward progression. During the swing phase, when one leg is in the air, swinging forward, the leg itself behaves as a pendulum, exchanging potential and kinetic energy in its forward arc with similarly high efficiency. The net metabolic cost of walking at comfortable speeds is therefore much lower than that of moving the body's mass and the limbs' masses against gravity would suggest. At comfortable walking speeds, about 65% of the total energy used in a step is recovered through these pendular mechanisms, compared to essentially zero recovery during running, where the bouncing spring mechanism provides different savings. The muscles work hard to initiate each step and to redirect the body's center of mass at the transitions. But between these transitions, the body's own physics carries much of the load. The ability of skeletal muscle to generate heat, thermogenesis, is one of its most important physiological functions in warm-blooded animals, and in conditions of cold stress the skeletal muscles can increase heat production by up to 5 times basal levels through shivering. The rapid, oscillating contractions of multiple muscle groups that generate heat without productive mechanical work. The muscle is the body's primary furnace. In an adult human at rest, approximately 25% of the body's total heat production comes from skeletal muscle. Even at rest, simply from the inefficiency of cross-bridge cycling and the cost of maintaining resting membrane potential and calcium homeostasis. During exercise, the fraction rises dramatically. At maximal exercise intensity, skeletal muscle accounts for more than 90% of total body heat production, the metabolic rate of the working muscles being so much greater than that of all other tissues combined. In cold conditions, when the body's core temperature threatens to fall below the thermoregulatory set point, the skeletal muscles are recruited specifically for heat production through shivering. Shivering is a pattern of rhythmic, oscillating muscle contractions, typically at 3 to 7 cycles per second, distributed across multiple muscle groups but often most visible in the neck, shoulders, and trunk. The contractions are not coordinated to produce useful mechanical work. Shivering is not a set of functional movements, but rather they are timed to be largely isometric or to cancel each other out, so that the ATP consumed in cross-bridge cycling is converted almost entirely to heat rather than mechanical work. Shivering can increase the body's metabolic rate by up to 5 times basal, producing a corresponding increase in heat generation that can sustain core temperature in cold environments for extended periods. The shivering reflex is controlled by the hypothalamus, which monitors core temperature and initiates shivering when the temperature falls below the set point. The skeletal muscle is not merely a movement machine. It is the body's thermostat, capable of turning up the heat on demand. Through the metabolic inefficiency it can amplify when necessary. The psoas major, The deep hip flexor muscle that runs from the lateral surfaces of the lumbar vertebrae to the lesser trochanter of the femur is unique among muscles in that it is simultaneously a muscle of the spine and a muscle of the hip, capable of both flexing the hip and contributing to lumbar stability, and its tension has been implicated in the common presentation of lower back pain. The psoas major arises from the bodies of T12 through L5 and the intervertebral discs between them, descending laterally through the abdomen alongside the vertebral column, passing beneath the inguinal ligament, and inserting on the lesser trochanter of the femur. It is a deep muscle, completely inaccessible to direct palpation in most people, lying anterior to the lumbar vertebrae and posterior to the abdominal viscera, and its architectural position gives it a mechanical relationship with the lumbar spine that is unlike any other limb muscle. At the hip, it is a powerful flexor and external rotator, firing strongly with every step forward and in every activity that involves bringing the thigh toward the trunk. But its proximal attachments to the lumbar vertebrae mean that it also generates a compressive and lordousing force on the lumbar spine when it contracts. It pulls the lumbar vertebrae toward the femur, increasing the lumbar lordosis and compressing the lumbar discs. In people who spend many hours seated, and modern sedentary life is characterized by extended periods of hip flexion that keeps the psoas in a shortened position. The psoas can develop adaptive shortening and sustained resting tone that maintains an anterior tilt of the pelvis and an excessive lumbar lordosis, loading the posterior elements of the lumbar spine and potentially contributing to the lower back pain that has become one of the most common musculoskeletal complaints in the contemporary world. Stretching the hip flexors, strengthening the gluteal muscles that oppose the psoas's pelvic tilt, and reducing the hours of seated hip flexion that the psoas is asked to maintain. These are the rehabilitative strategies that address the muscular component of lower back pain, because they address the muscle that has been sitting at the heart of the problem all along. The muscle fiber's membrane potential, the electrical difference between the interior and exterior of the fiber at rest, is approximately -70 to -90 millivolts. Maintained by the differential distribution of sodium, potassium, and chloride ions across the membrane and by the continuous activity of the sodium-potassium ATPase pump. The resting membrane potential of the skeletal muscle fiber is the electrical baseline from which every action potential departs. The stable, electrically polarized state that the muscle maintains between stimulations, representing the ready state of the excitable membrane. It is created by the differential distribution of ions across the sarcolemma, a high internal potassium concentration and a high external sodium concentration, maintained against the diffusive tendency that would otherwise equalize them across the membrane by the continuous activity of the sodium-potassium ATPase. This pump, using one ATP molecule per cycle, transports 3 sodium ions outward and 2 potassium ions inward, maintaining the gradients that are the electrochemical basis of the resting potential. Because the resting membrane is much more permeable to potassium than to sodium, through constitutively open potassium leak channels, potassium ions diffuse outward down their concentration gradient, carrying positive charge out of the cell and leaving behind unpaired negative charges inside, creating the negative interior potential. The magnitude of the resting potential reflects the balance between the tendency of potassium to diffuse out, which hyperpolarizes the membrane, and the modest inward diffusion of sodium, which depolarizes it slightly. The Nernst potential for potassium, the potential at which the electrical gradient exactly opposes the concentration gradient for potassium, is approximately -90 millivolts, and the resting potential of most excitable cells sits near this value, modified by the small depolarizing influence of sodium conductance. Every action potential that has ever activated one of your muscles began at this potential, at the charged, ready, waiting electrical state of the resting sarcolemma. Tonight, the muscle fibers are holding this potential in silence, doing no other electrical work, simply being ready. A second question. I wonder how you are feeling right now. Whether the day that seemed so large when you arrived here tonight has shrunk at all. Whether the muscles of the jaw and the shoulders and the space between the eyebrows have found a little more release than they had at the beginning of this. If they have, even a degree, I am glad. And if you feel like leaving something in the comments about what tonight feels like from where you are, I would genuinely love to read it. You do not need to say much. Just, I was here. This helped. That is enough. The concept of muscle tone, the slight, continuous partial contraction that healthy muscle maintains at rest, is not due to a low level of active cross-bridge cycling but primarily to the passive elastic properties of the muscle and connective tissue, with a small active component from a low baseline of motor neuron activity that varies by muscle and postural demand. Muscle tone. What a clinician assesses when they flex and extend a patient's limb passively and feel the resistance is a composite of several things that are conceptually distinct but physically inseparable in the clinical examination. The dominant component, especially in relaxed muscle, is passive stiffness, the resistance to deformation provided by the elastic properties of the titin, the connective tissue, the sarcolemma, and the other non-contractile structural elements of the muscle and its surrounding fascia. This passive stiffness does not require any neural activity and does not consume metabolic energy. It is a material property of the tissues, present even in completely denervated muscle. The active component, the actual low-level firing of motor neurons driving a small fraction of motor units, contributes to tone and postural muscles that are tonically active, but in fully relaxed muscle at rest, particularly in large limb muscles with the person lying down. This active component is minimal. The distinction between the passive and active components of muscle tone has clinical importance because certain neurological conditions affect them differentially. Spasticity, the velocity-dependent increase in tone seen after upper motor neuron lesions such as stroke or spinal cord injury, involves an abnormal increase in the active component, specifically an exaggerated tonic stretch reflex driven by the release of the spinal motor neurons from descending inhibitory control. Rigidity, the lead pipe or cogwheel resistance seen in Parkinson's disease, involves an increase in both active and passive components, The active component driven by the enhanced excitability of alpha motor neurons due to loss of dopaminergic modulation. Hypotonia, the floppy tone of lower motor neuron lesions or cerebellar disease, involves a reduction in the active component and sometimes in the passive properties as well. Tone is not simple. It is a conversation between the passive properties of tissue and the active contributions of the nervous system. The muscles of the hand, both the intrinsic muscles within the hand itself and the extrinsic muscles in the forearm whose long tendons cross into the hand, must coordinate their activity with extraordinary precision to produce the full range of hand function, from the power grip of lifting a heavy object to the precision pinch of threading a needle. The hand's remarkable functional range, encompassing power grips that can sustain hundreds of newtons, precision pinches that can manipulate objects of a millimeter, requires a muscular architecture of corresponding complexity. The intrinsic muscles of the hand, the thenar muscles at the thumb base, the hypothenar muscles at the little finger base, the 4 dorsal and 3 palmar interossei, and the 4 lumbricals, provide the fine-grained control of finger position and the coordinated force patterns of precision movements. The lumbricals, in particular, have a unique architecture. They originate from the tendons of the flexor digitorum profundus and insert into the extensor hood of the finger, allowing them to simultaneously flex the metacarpophalangeal joint and extend the interphalangeal joints, the intrinsic plus position that is optimal for fine manipulation. The interossei abduct and adduct the fingers and also contribute to this intrinsic plus position. The extrinsic muscles, the flexors in the forearm whose tendons run through the carpal tunnel to the finger flexors, and the extensors on the dorsum, provide the power for large gripping forces and the extension necessary for releasing objects. The coordination of intrinsic and extrinsic muscles in tasks like piano playing, surgical instrument handling, or the delicate positioning required for written script represents one of the most demanding feats of neuromuscular coordination in human motor behavior. The hand's cortical motor representation in the primary motor cortex is disproportionately large relative to the hand's physical size. The cortical homunculus devotes approximately as much area to the hand as to the entire trunk, reflecting the neural investment required to control this system with the precision that human hand function demands. The smooth muscle of the airway walls, the bronchial smooth muscle, can contract in response to various stimuli including cold air, exercise, allergens, and parasympathetic activation, narrowing the lumen of the bronchi and bronchioles and increasing airway resistance in a process that, when it occurs excessively, produces the wheezing and breathlessness of asthma. In the healthy airway, the bronchial smooth muscle maintains a resting tone that contributes a modest amount of resistance to airflow. Not trivial but not clinically significant. The smooth muscle is innervated by both parasympathetic fibers, which drive bronchoconstriction, and sympathetic fibers, which drive bronchodilation, from the autonomic nervous system, and it expresses receptors for a wide range of locally and systemically produced mediators. Exercise normally produces bronchodilation. The sympathetic activation and circulating adrenaline of exercise activate the beta-2 adrenergic receptors on the bronchial smooth muscle, causing relaxation and widening of the airways to accommodate the increased ventilatory demand. Cold air can cause bronchoconstriction in susceptible individuals. The mechanism is not entirely clear but appears to involve both direct cooling effects on the smooth muscle and reflex activation through the trigeminal nerve. In asthma, the bronchial smooth muscle is hyperresponsive. Its threshold for constriction is lower than normal, and stimuli that produce modest, transient narrowing in a healthy airway produce sustained, severe narrowing in an asthmatic airway. This hyperresponsiveness is partly the intrinsic property of the smooth muscle cells themselves, which are enlarged and produce more contractile force per cell in the asthmatic airway, and partly the result of the inflammatory environment of the asthmatic airway. Mast cells, eosinophils, and their mediators sensitize the smooth muscle and lower its threshold for constriction. The treatment of asthma exploits the airway smooth muscles pharmacology directly. The beta-2 agonist bronchodilators, salbutamol, formoterol, activate the same adrenergic receptors that sympathetic activity uses to relax the bronchial smooth muscle, overriding the constriction with pharmacological relaxation. The muscle of the ciliary body, the ciliary muscle, is a smooth muscle that controls the shape of the crystalline lens of the eye, allowing the eye to change its focal length for near and far vision in the process called accommodation. And its gradual weakening with age produces the presbyopia that affects most people by their mid-40s. The crystalline lens of the eye is remarkable not just for its transparency, a property maintained by the precise, highly ordered arrangement of its constituent crystalline proteins, but for its elasticity, which allows its shape to be changed by the action of the surrounding ciliary muscle. In the young eye, the lens is soft and pliable, held in place by the ciliary zonial fibers that run from its equator to the ciliary body. In the relaxed state, when the ciliary muscle is relaxed and the zonial fibers are taut, the lens is stretched to a relatively flat shape by the zonial tension, and the eye is focused for distance. When the ciliary muscle contracts, it is a ring of smooth muscle, and its contraction reduces the internal diameter of the ring. The tension on the zonial fibers is released, and the elastic lens rounds up, its surfaces becoming more convex and its optical power increasing, shifting the focal point closer to focus on near objects. This active muscular accommodation, the ability to shift focus smoothly from far to near by modulating ciliary muscle contraction, is one of the optical capabilities that makes the eye so versatile. Presbyopia, the progressive loss of accommodation that occurs with advancing age, is caused by the progressive hardening of the lens, not by weakening of the ciliary muscle. The lens accumulates new fibers throughout life and loses its suppleness, becoming increasingly resistant to the shape change that ciliary muscle contraction would otherwise produce. By the mid-40s, the lens is stiff enough that even maximal ciliary muscle contraction cannot produce the rounding required for near focus without optical assistance. The reading glasses that appear in most people's lives in their 40s are the compensation for a lens that has simply lost the flexibility it once had. The muscles of inspiration are assisted during exercise by a phenomenon called the respiratory muscle metaboreflex, an increase in sympathetic vasoconstriction to the locomotor muscles triggered by accumulation of metabolic byproducts in the respiratory muscles when their blood supply becomes insufficient, which can limit exercise performance when the respiratory muscles compete with the locomotor muscles for blood flow. At very high exercise intensities, the respiratory muscles, primarily the diaphragm and the external intercostals, begin to work extremely hard, their metabolic demand rising sharply as ventilation rates approach and exceed 100 liters per minute. To sustain this high metabolic rate, the respiratory muscles need a proportionally increased blood flow. But the cardiovascular system, already near its maximum output, may not be able to increase flow to the respiratory muscles without reducing flow to the already highly active locomotor muscles of the legs. When the respiratory muscles become relatively ischemic, when their blood supply is insufficient for their metabolic rate, group 3 and 4 afferent fibers in the diaphragm and intercostals begin to discharge, signaling the accumulating metabolic byproducts. This afferent discharge activates sympathetic vasoconstriction reflexively, reducing blood flow to the locomotor muscles to redirect blood to the respiratory muscles. The consequence for the exercising athlete is a reduction in locomotor muscle perfusion at exactly the moment of maximum effort, limiting the delivery of oxygen and the removal of metabolites from the working legs. Studies using inspiratory muscle training, specific conditioning of the respiratory muscles to increase their endurance and reduce the degree of ischemia they experience at high ventilation rates, have shown modest but real improvements in endurance performance, consistent with the reduction of this respiratory muscle metaboreflex. The respiratory muscles, in other words, do not merely support exercise. They can, at the extreme, compete with the locomotor muscles for the resources that performance demands. The lungs and the legs are not always on the same side. The microscopic architecture of the muscle fiber reveals a stunning degree of spatial organization. The myofibrils, mitochondria, sarcoplasmic reticulum, T-tubules, and glycogen granules are arranged in precise, reproducible relationships to each other that optimize the diffusion distances for calcium, oxygen, and metabolic substrates. Looking at a skeletal muscle fiber under the electron microscope is, if you understand what you are seeing, one of the more visually compelling experiences in biology. The longitudinal sections reveal the repeating sarcomere pattern, the Z discs and A bands and I bands in precise parallel arrays, running along the full length of myofibrils that are themselves packed side by side with a regularity that approaches crystalline. Between the myofibrils, the sarcoplasmic reticulum forms a continuous membranous network, the longitudinal SR tubules running along the A bands and the terminal cisternae bulging at each end of the sarcomere, flanking the T-tubule that dives inward from the sarcolemma. The triad, one T-tubule flanked by two terminal cisternae, appears at each AI junction, positioned precisely where the calcium release must occur to activate the adjacent sarcomere. Mitochondria are positioned immediately adjacent to the myofibrils, often in longitudinal rows between them, minimizing the diffusion distance for the ATP that the mitochondria produce and the ADP and inorganic phosphate that must return to them for reprocessing. In fast-twitch fibers, where ATP demand is high but oxidative capacity is lower, glycogen granules are packed between the myofibrils as the immediate glycolytic fuel reserve. In slow-twitch fibers, the mitochondria are more numerous, more closely opposed to the contractile apparatus, and the capillary density surrounding the fiber is higher, minimizing the diffusion distance for oxygen from the blood. Every aspect of this spatial organization reduces the time and distance over which metabolic signals and substrates must travel. The spatial organization is itself an optimization, ensuring that the molecular machinery of contraction and energy production are as close together as physics and cell biology will allow. The extensor digitorum longus and the tibialis anterior, muscles on the front of the lower leg, are the primary dorsiflexors of the foot and the muscles responsible for lifting the foot clear of the ground during the swing phase of gait, and their weakness or paralysis produces the characteristic gait disturbance called foot drop. Foot drop, the inability to adequately dorsiflex the foot during the swing phase of walking, is one of the more visually distinctive gait disorders, and it arises from weakness or paralysis of the ankle dorsiflexors, primarily the tibialis anterior. In normal walking, the tibialis anterior contracts concentrically immediately after heel strike to control the rate of foot lowering to the ground, preventing the foot from slapping down, and then contracts again during the swing phase to lift the toes and forefoot clear of the ground. Without this swing phase dorsiflexion, the foot hangs in plantar flexion during swing, and if not compensated by circumducting the hip, swinging the leg outward in a wide arc to provide clearance, or by excessive hip flexion, steppage gait, the toes would drag on the floor with each step. The most common cause of foot drop is common peroneal nerve injury. The nerve, a branch of the sciatic, wraps around the neck of the fibula where it is vulnerable to compression from prolonged crossing of the legs, to external pressure during unconsciousness or anesthesia, and to direct trauma. Less commonly, foot drop results from lesions of the L4 or L5 nerve roots, from fibula head fractures, or from central neurological conditions affecting the corticospinal control of the anterior compartment muscles. The tibialis anterior is easily tested by asking a patient to dorsiflex the foot against resistance, and its weakness is one of the most helpful localizing signs in neurological examination of the lower limb. The muscle that lifts the foot with each step, unremarkable in its function when it is working, immediately conspicuous in its absence. The relationship between muscle architecture and the range of motion it can produce is governed by the principle that a muscle with longer fibers can shorten through a greater absolute distance, and that muscles with longer fibers therefore produce greater range of joint motion than muscles with shorter fibers of equal physiological cross-sectional area. If force is proportional to physiological cross-sectional area and range of motion is proportional to fiber length, then the architecture of a muscle, the combination of its fiber length and its pennation angle, determines the combination of force and range of motion it can provide for a given volume of muscle tissue. Long fibers provide range of motion, but for a given muscle volume, mean fewer fibers in parallel and therefore less force-generating area. Short, heavily pennate fibers provide force but at the cost of range of motion. Real muscles in the body fall at different positions along this force-velocity range trade-off depending on their functional requirements. The hamstrings, biceps femoris, semimembranosus, semitendinosus, have relatively long fibers and span two joints, giving them the ability to produce motion across a large range of knee flexion and hip extension angles. They are important for acceleration and sprinting, which require force production across large ranges of motion. The gastrocnemius has moderate fiber length and moderate pennation. It can produce substantial plantar flexion force across a reasonable range of ankle motion. The soleus is more heavily pennate, with shorter fibers and greater physiological cross-sectional area relative to volume. It trades some range of motion for greater force-generating capacity, appropriate for its role in sustaining the large forces of standing and walking at relatively constant ankle angles. These architectural decisions are evolutionary optimizations. Each muscle is shaped by the specific combination of force and motion requirements of its function, arriving at the architecture that best satisfies both constraints given the volume available. The body's largest muscle by volume, not the gluteus maximus, which is largest by mass, but the quadriceps femoris as a functional group, can generate extension torques at the knee of more than 300 Newton-meters in trained athletes, exceeding the mechanical strength of the patellar tendon under certain conditions and explaining the occasional catastrophic tendon ruptures seen in maximal efforts. The quadriceps femoris is a group of four muscles: the rectus femoris, vastus lateralis, vastus medialis, and vastus intermedius, that converge on the patella through the quadriceps tendon and then extend to the tibial tuberosity through the patellar tendon. Together, they represent the largest single-function muscle group in the body, their combined volume and physiological cross-sectional area exceeding that of any other muscle group. The maximum knee extension torque this group can generate in a well-trained athlete, during a maximal isometric contraction at the optimal knee angle of approximately 70 degrees of flexion, can exceed 300 Newton-meters. The patellar tendon, which transmits this torque to the tibia, is typically stressed to approximately 30 to 40 megapascals during this effort, a significant fraction of its tensile strength, typically reported at 50 to 100 megapascals. During explosive maximal efforts, a heavy landing, a maximal vertical jump, the forces on the patellar tendon can transiently exceed these static values, approaching the tendon's failure stress. Patellar tendon rupture during athletic activity, though uncommon, is not rare, and it occurs precisely in these moments of explosive maximal effort. The tendon is designed to survive the forces of ordinary activity with a large safety factor, but the extraordinary forces of maximal athletic effort narrow this factor considerably. The patellar tendon that ruptures during a basketball player's explosive jump was not, in most cases, a normal tendon. Chronic overuse typically produces the tendinopathic changes that reduce its strength before the final catastrophic rupture. But the force that delivers the final insult is the quadriceps group, delivering its maximum output through a structure that, on that particular day, was not quite equal to the task. The muscle's oxygen store, bound to the myoglobin that gives red muscle its color, plays an important role in sustaining oxidative metabolism during brief interruptions to blood flow, such as occur during high-force contractions when the elevated intramuscular pressure compresses the capillaries and temporarily occludes blood flow to the contracting fibers. Myoglobin is a single-chain, iron-containing protein that binds oxygen with approximately 10 times the affinity of hemoglobin. Its oxygen dissociation curve is not sigmoidal like hemoglobin's but hyperbolic. In its P50, the partial pressure at which it is 50% saturated is approximately 5 millimeters of mercury, compared to hemoglobin's 26. This very high oxygen affinity means that myoglobin holds onto its oxygen at the low partial pressures that exist in actively metabolizing muscle, releasing it only when the local oxygen tension falls below approximately 5 millimeters of mercury. Under these conditions, myoglobin serves as an oxygen buffer, a store that releases oxygen to the mitochondria when supply from the blood is momentarily insufficient. High-force isometric contractions generate intramuscular pressures that can exceed the perfusion pressure of the capillaries, compressing them and temporarily stopping blood flow to the contracting fibers. During these periods, which may last for the duration of the contraction, several seconds, the oxygen stored in myoglobin sustains oxidative phosphorylation in the mitochondria without interruption, delaying the onset of anaerobic metabolism and the associated metabolic acidosis. The deep red color of slow-twitch muscle compared to the pale color of fast-twitch muscle reflects the higher myoglobin content of slow fibers, fibers that rely predominantly on oxidative metabolism and that contain more of the oxygen-storing protein that supports it. The myoglobin in the soleus is doing this right now, holding its oxygen quietly, ready to release it in the next bout of contractions when the blood supply is momentarily compressed by the contraction itself. Even the oxygen store is organized in muscle for efficiency. The cardiac muscle's response to increased filling, the Frank-Starling mechanism, ensures that the heart automatically matches its output to the venous return without requiring any neural adjustment, through the increase in contractile force that accompanies stretching of the myocardium beyond its resting length. The Frank-Starling law of the heart, named for Otto Frank and Ernest Starling, who described its components in the late 19th and early 20th centuries, is one of the most important intrinsic regulatory mechanisms of cardiac function. It states that, within physiological limits, the strength of cardiac muscle contraction increases as the initial length of the muscle fibers increases, or, expressed at the whole heart level, that stroke volume increases as end diastolic volume increases. The mechanism underlying this response was, for many decades, attributed simply to the length-tension relationship of the sarcomere, the greater force at optimal sarcomere overlap described by the length-tension curve. This explanation is correct in part, but it does not fully account for the magnitude of the Frank-Starling response. A larger component comes from the increase in calcium sensitivity of the contractile apparatus with stretch. At longer sarcomere lengths, the calcium sensitivity of troponin C increases, so that the same calcium transient produces more cross-bridge formation and more force. Part of this calcium sensitivity increase results from the geometric changes at longer sarcomere lengths. The closer proximity of the myosin heads to the actin filaments reduces the lattice spacing of the myofilaments and increases the probability of cross-bridge formation. The Frank-Starling mechanism operates continuously to match the heart's output to the venous return if more blood returns to the right atrium because of increased activity or increased venous tone. The ventricle is filled to a greater end diastolic volume, stretched to a longer fiber length, and contracts more forcefully, ejecting the additional volume. No neural signal is required. The heart adjusts itself automatically through the inherent properties of the cardiac muscle fibers. The proprioceptive sense, the sense of limb position and movement provided by muscle spindles, Golgi tendon organs, and joint receptors, is essential for skilled motor control, and its disruption by peripheral neuropathy or posterior column spinal cord disease produces the characteristic sensory ataxia in which movements are uncoordinated despite intact motor function and adequate muscle strength. Proprioception is the sense you most take for granted and would most immediately miss. With eyes closed, in a dark room, you know exactly where your limbs are, the angle of each joint, the tension in each muscle, the position of each finger. You can reach for a glass in the dark, button a shirt without looking, write your signature with your eyes closed and produce something essentially identical to what you write with them open. All of this depends on the continuous stream of proprioceptive information from the muscles and joints to the somatosensory cortex and the cerebellum. When this stream is disrupted by peripheral neuropathy damaging the large diameter sensory fibers that carry proprioceptive signals, or by damage to the posterior columns of the spinal cord through which those signals ascend, the result is sensory ataxia. The affected individual, with eyes open and in good light, may function almost normally. The visual system compensates for the lost proprioceptive input, providing a slower but functional replacement. But with eyes closed, or in the dark, or whenever the visual system is distracted by other demands, the ataxia becomes manifest, an inability to maintain posture without visual reference, the positive Romberg sign, an unsteady wide-based gait, a tendency for the unsupported limb to drift from its intended position. The hands fumble with buttons because they cannot feel where they are relative to the fabric. The movements are strong, the muscles are intact, but directionless, because the position sense that normally guides them is absent. Proprioception, the muscle's continuous report to the brain of where the body is not merely a background feature of motor control. It is the navigational system without which motor control loses its way. The muscles used in breathing do not rest between breaths as most voluntary muscles rest between contractions. Instead, the expiratory phase of quiet breathing involves the passive recoil of the respiratory system. but the inspiratory muscles must begin their next contraction before expiration is complete, in a pattern of continuous, overlapping activity that ensures smooth, uninterrupted ventilation. The respiratory muscles operate under constraints that no other muscle group faces. They must produce continuous rhythmic contractions without any sustained rest period, across the full range of body states from deep sleep to maximal exercise, without the option of taking a break when fatigued. The strategy that the respiratory control system uses to accommodate this is the overlap of inspiratory and expiratory muscle activity. The inspiratory muscles do not fully relax during expiration but instead progressively reduce their activity, breaking the speed of expiration and preventing the lung from collapsing to its minimum volume at the end of each breath. The smooth sinusoidal pattern of the normal breathing cycle, gradual inhalation, smooth exhalation, brief pause, belies the continuous muscular activity that underlies it. The diaphragm and external intercostals begin to increase their activity approximately 50 milliseconds before the onset of inspiratory airflow, pre-activating to overcome the inertia of the respiratory system. Their activity peaks at the moment of peak inspiratory flow and then decreases, the decreasing activity during the first part of expiration breaking the outward elastic recoil of the lungs and chest wall, controlling the rate of expiration. Only when expiratory flow has largely ceased does the inspiratory muscle activity fall to its interbreath minimum. And then, within a fraction of a second, the next inspiratory effort begins. The respiratory muscles are never truly off. They are always in some phase of this continuous cycle, braking or accelerating, active or minimally active, never fully at rest. Even now, as you lie in the quiet dark, your diaphragm is cycling. It will cycle all night. It will cycle for the rest of your life. The phenomenon of tetanus, the sustained, maximal contraction produced by a rapid series of stimuli that prevents the muscle from relaxing between stimuli, is the contractile state that produces maximum force, and the transition from single twitches to unfused tetanus to fused tetanus as stimulus frequency increases explains the graded force production of muscle in response to varying neural firing rates. A single action potential in a motor neuron produces a single twitch in the muscle fibers of its motor unit, a brief, transient contraction that rises to a peak force in approximately 50 to 100 milliseconds and then declines over a similar period as calcium is resequestered and cross-bridges detach. The peak force of a single twitch is only about 30 to 40% of the maximum force the fiber can produce, because the calcium transient is brief and not all available binding sites on troponin are occupied simultaneously. If a second stimulus arrives before the twitch has fully relaxed, the calcium from the second stimulus adds to the residual calcium from the first, and the force of the second twitch summates with the residual force of the first. Temporal summation. At low stimulus frequencies, the twitches summate partially, producing an unfused tetanus, a series of peaks and valleys in the force record, each peak higher than the preceding one until a plateau is reached. At higher stimulus frequencies, typically above 50 to 80 Hz for fast fibers, The twitches fuse completely. The sarcoplasmic reticulum cannot resequester calcium between stimuli. Intracellular calcium remains elevated throughout, and the force reaches and holds at the titanic maximum, the maximum isometric force the fiber can develop, substantially greater than the single twitch force. The motor system uses this frequency-force relationship in reverse. By varying the firing rate of motor neurons between the threshold for activation and the fusion frequency, it can vary the force of each motor unit continuously from near zero to maximum, providing the graded force control that underlies the smooth modulation of voluntary movement force. The muscle spindle primary endings, the IA afferents, project not only to the spinal cord but directly to the cerebellum through spinocerebellar tracts, providing the cerebellum with a continuous, real-time stream of proprioceptive information that is used to update the cerebellum's internal models of limb mechanics and to detect discrepancies between the intended and actual movements. The cerebellum, the cauliflower-shaped structure at the back of the brain, containing more neurons than the rest of the brain combined, is the body's motor calibration system. It compares the motor commands sent out from the motor cortex, the efferent's copy, with the sensory feedback arriving from the periphery, the proprioceptive signals from muscle spindles, Golgi tendon organs, and joint receptors, computes the difference, and uses this error signal to generate corrective inputs to the motor cortex and brainstem motor nuclei. The speed and accuracy of this comparison depends critically on the rapid delivery of proprioceptive information to the cerebellum and the spinocerebellar tracts, several parallel pathways running from the spinal cord directly to the cerebellum, without synapsing in the thalamus or cortex, are the fastest sensory pathways in the body, conducting at velocities up to 120 meters per second. The IA afferents from muscle spindles enter the spinal cord and, in addition to their monosynaptic connections to the alpha motor neurons of the same muscle, send collateral branches into the spinocerebellar tracts. The cerebellum thus receives, within milliseconds of any change in muscle length, information about what the muscles are doing. This real-time proprioceptive input is what allows the cerebellum to track the progress of a movement as it is occurring, to detect when it is deviating from the intended trajectory, and to issue corrections that arrive at the motor neurons fast enough to influence the ongoing movement. Cerebellar damage disrupts this real-time tracking, producing the characteristic intention tremor and dysmetria of cerebellar ataxia, the overshooting and undershooting of movement targets that results when the calibration system loses its proprioceptive input. The contractile proteins of muscle, actin, myosin, titin, tropomyosin, troponin, account for approximately 70 to 80% of the total protein content of a muscle fiber, with myosin alone constituting approximately 25 to 30%, making the sarcomere one of the most protein-dense structures in the cell. The interior of the skeletal muscle fiber is, at steady state, packed to a degree that makes it one of the most protein-concentrated compartments in the body. The myofibril, which occupies the majority of the fiber's volume, contains within each cubic micrometer of sarcomere hundreds of myosin molecules and thick filaments, hundreds more actin molecules and thin filaments, tighten molecules spanning the full half sarcomere and all the regulatory proteins associated with them. The concentration of myosin in the A band of the sarcomere exceeds 300 milligrams per milliliter, approximately 10 times the total protein concentration of most eukaryotic cytoplasm. This extraordinary packing density is possible because the proteins are organized into the crystalline-like regularity of the myofilament lattice, which allows proteins to be packed at near maximum density while maintaining the precise geometric relationships required for cross-bridge function. Myosin alone, at approximately 25 to 30% of total protein, represents a metabolic investment of remarkable magnitude. Each myosin molecule takes days to synthesize and assemble, and the total amount of myosin in the human skeletal muscle mass represents one of the largest single protein investments in the body. This is why protein synthesis is a primary limiting factor in muscle hypertrophy. The building of additional sarcomeres requires the production of large quantities of myosin and actin, a process that demands adequate dietary protein, adequate anabolic signaling, in sufficient time. The sarcomere is, in its biochemical economy, one of the most expensive structures the body maintains. Its expense is also its capability. Every kilogram of muscle protein represents a store of force-generating potential that can be called upon whenever the body requires it. The muscle of the bladder wall, the detrusor, is a smooth muscle with the unusual property of being able to accommodate very large changes in volume, from 30 milliliters to 500 milliliters or more, with minimal change in intravesical pressure, through a combination of mechanical compliance and active muscular relaxation under inhibitory neural control. The detrusor muscle's ability to store progressively larger volumes of urine without generating a significant increase in pressure, the property called bladder compliance, is essential to normal urinary function. A bladder that responded to filling with progressively increasing pressure would generate urgency at low volumes, be unable to store adequate amounts between voidings, and would transmit elevated pressures to the ureters and kidneys, potentially damaging the upper urinary tract. Normal compliance allows the bladder to fill from its minimal volume to its comfortable capacity of 300 to 500 milliliters with an intravesical pressure rise of only 10 to 15 centimeters of water, a remarkably small increase for such a large volume change. This compliance comes from two sources. The passive component, the viscoelastic properties of the smooth muscle and connective tissue of the bladder wall, contributes substantially, the wall stretching and accommodating the increasing volume through the mechanical properties of its fibers. The active component, inhibition of detrusor contraction through the sympathetic hypogastric nerve, which relaxes the smooth muscle, maintains the muscle in a non-contracting, compliant state during filling. The sensation of bladder filling, and the transition from comfortable fullness to urgency, is mediated by mechanoreceptors and chemoreceptors in the bladder wall whose afferent signals travel to the pontine micturition center and the periaqueductal gray, the areas of the brainstem that coordinate the micturition reflex. When voiding is appropriate, The pontine micturition center coordinates the simultaneous relaxation of the external urethral sphincter and the contraction of the detrusor, a carefully timed sequence that requires the inhibition of the sphincter and the facilitation of the detrusor in precise coordination. The muscle's response to heat stress. The production of heat shock proteins in response to elevated temperature, oxidative stress, or intense exercise is one of the most highly conserved stress response mechanisms in biology, and the heat shock proteins they encode serve as molecular chaperones that protect the contractile proteins from denaturation and facilitate the repair of damaged proteins. Heat shock proteins, so named because they were first discovered in response to thermal stress in Drosophila cells, are a family of molecular chaperones that are induced in response to a wide range of cellular stresses, including not just heat but oxidative stress, hypoxia, mechanical stress, and the metabolic perturbations of intense exercise. In muscle cells, exercise-induced heat shock protein expression, particularly the induction of HSP70, HSP90, and the small heat shock proteins including alpha-B-crystalline, serves multiple protective and reparative functions. Molecular chaperones assist in the correct folding of newly synthesized proteins, preventing the aggregation of misfolded chains that would otherwise form toxic protein aggregates within the cell. They recognize and bind to proteins that have been partially denatured by the heat and oxidative stress of intense exercise, stabilizing them against further unfolding and facilitating their refolding to the native state when conditions improve. Some heat shock proteins target irreparably damaged proteins for degradation by the proteasome, removing them before they can interfere with cellular function. The induction of heat shock proteins by exercise is part of the same cellular adaptation response that produces the increased tolerance to exercise-induced stress seen with training. Muscles that have been repeatedly exercised express more constitutive heat shock proteins and mount a faster and larger inducible response to subsequent stress, making the contractile proteins more resilient to the conditions of strenuous activity. The molecular chaperones that protect the muscle's proteins are themselves produced by the stresses that damage those proteins. The protection grows from the challenge. The muscles of the thoracic and abdominal wall work in coordination with the diaphragm and the pelvic floor to generate and control the intra-abdominal pressure that is used for Valsalva maneuvers, defecation, childbirth, and the bracing of the lumbar spine during heavy lifting, a pressure that can exceed 200 millimeters of mercury in trained lifters. The Valsalva maneuver, a forced expiratory effort against a closed glottis, is the mechanism by which the thoracic and abdominal muscles collaborate to generate the highest intra-abdominal pressures the body can produce. The sequence is inspiration, closure of the glottis by the laryngeal muscles, then contraction of the diaphragm and all the muscles of the abdominal wall simultaneously. The diaphragm descends. The abdominal muscles compress the abdominal contents from all sides. The glottis prevents any gas escape, and the pressure within the closed abdominothoracic cavity rises. The resulting intra-abdominal pressure stiffens the lumbar spine through the mechanism of the hydraulic amplifier. The pressurized abdominal cavity acts like an inflated balloon supporting the lumbar vertebrae from the front, reducing the bending moment that the paraspinal muscles must counteract. In trained powerlifters performing maximal deadlifts and squats, intra-abdominal pressures exceeding 200 millimeters of mercury, substantially above the normal systolic blood pressure, are recorded during the lift. These pressures are tolerated because the simultaneously elevated thoracic pressure reduces the transmural pressure, the pressure difference across the vessel wall of the great veins in the heart, preventing the cardiovascular complications that such pressures would otherwise produce. The same mechanism, coordinated muscle contraction generating intra-abdominal pressure, is used in defecation, in childbirth, in heavy lifting, and in coughing. It is one of the body's most powerful collaborative muscular efforts, bringing together the diaphragm, the abdominal wall, the pelvic floor, and the larynx in a single, coordinated increase in contained pressure. The oxygen consumption of resting muscle is approximately 2 to 3 milliliters of oxygen per 100 grams per minute, rising to 60 to 70 milliliters per 100 grams per minute during maximal exercise, a 30-fold increase that requires every component of the oxygen delivery and utilization chain to function at or near maximum capacity simultaneously. The 30-fold increase in muscle oxygen consumption from rest to maximal exercise is one of the most dramatic metabolic transitions in biology, and its achievement in under a minute is a demonstration of the remarkable coordination of the cardiovascular, respiratory, and metabolic systems. At rest, the skeletal muscle extracts approximately 25 to 30% of the oxygen delivered to it by the blood, a modest extraction fraction, leaving a large reserve for times of increased demand. At maximal exercise, this fraction rises to approximately 80 to 85%. The muscle is extracting nearly all the available oxygen, and the venous blood leaving the working muscle contains very little O2. Simultaneously, the delivery of oxygen to the muscle has increased through the combination of increased cardiac output and the redistribution of blood flow to the active muscles. The combined effect of increased delivery and increased extraction produces the 30-fold increase in muscle oxygen consumption. The mitochondria, whose rate of oxidative phosphorylation sets the ceiling for oxygen consumption, are functioning at near maximal capacity. The ADP released from rapidly cycling crossbridges is the primary driver of mitochondrial activity, stimulating electron transport and ATP synthesis at rates that match the ATP demand of the contracting fiber. The oxygen, delivered to the mitochondria by myoglobin and the capillary blood, is consumed almost as fast as it arrives. The muscle at maximal exercise is operating at the very edge of its metabolic capacity, a state that can be sustained for only minutes, limited by the accumulation of metabolic byproducts and the progressive depletion of fuel substrates. The muscle's phospholipid membrane, the sarcolemma, is not a passive structural boundary but an active participant in signaling, containing mechanosensitive ion channels that open in response to membrane deformation during stretch and contraction, providing one of the mechanisms by which the muscle cell senses its own mechanical state. The sarcolemma's role as a mechanosensory interface connects the outside world, the mechanical forces applied to the muscle, with the inside of the cell, where the signaling cascades that regulate gene expression, protein synthesis, and cell survival are initiated. Mechanosensitive ion channels, including members of the piezo family and the transient receptor potential (TRP) channel family, are embedded in the sarcolemma and respond to the deformation of the membrane by changing their open probability. When the membrane is stretched, as occurs during eccentric contractions or during passive muscle stretch, these channels open, allowing calcium to enter the cell from the extracellular space. This mechanically triggered calcium entry is distinct from the calcium released from the sarcoplasmic reticulum during excitation-contraction coupling and is thought to contribute to the activation of downstream signaling pathways including the calcineurin NFAT pathway and the MAPK cascade. These pathways, in turn, regulate the expression of genes involved in muscle adaptation. The genes encoding contractile proteins, metabolic enzymes, and structural proteins whose expression must change as the muscle adapts to new mechanical demands. The mechanosensitive channels are part of the muscle's system for converting mechanical experience into biological response. The molecular transducers that allow the muscle to feel what is happening to it and to respond accordingly. The muscle is not merely a machine that generates force on demand. It is a mechanosensory organ that continuously reads the forces applied to it, translates them into molecular signals, and uses those signals to adjust its own properties over time. The muscle feels. In its way, it responds to what it has felt. And it becomes, gradually, what its experience has shaped it to be. The muscles of the middle ear, the stapedius and the tensor tympani, are the smallest skeletal muscles in the body and serve as active attenuators of loud sounds, contracting reflexively in response to self-generated sounds, including speech, and in anticipation of loud noises to reduce the transmission of sound energy through the ossicular chain, protecting the hair cells of the cochlea from acoustic trauma. The acoustic reflex, the reflex contraction of the stapedius and tensor tympani in response to loud sounds, is mediated by a pathway from the cochlea through the auditory brainstem nuclei to the motor nuclei of the facial and trigeminal nerves, which innervate the stapedius and tensor tympani respectively. The reflex latency is approximately 25 to 150 milliseconds depending on the sound level, fast enough to provide some protection against sustained loud sounds but too slow to protect against the sudden onset of an impulsive noise like a gunshot. The more interesting and more protective aspect of the acoustic reflex is its anticipatory component. Before a person speaks, the stapedius contracts about 25 milliseconds before the onset of voice, reducing the transmission of the person's own voice through the ossicular chain and preventing the masking of environmental sounds by one's own speech. A similar anticipatory contraction occurs before voluntary movement that is expected to generate loud sound, before striking a metal surface, before clapping. The anticipatory contraction is driven by corollary discharges from the motor cortex, copies of the motor commands for the anticipated action sent to the auditory system to pre-attenuate the expected sound. The two smallest muscles in the body are thus active participants in auditory perception, managing the sensitivity of the hearing apparatus in response to anticipated and actual loud sounds, protecting the irreplaceable hair cells of the cochlea by reducing the mechanical energy reaching them when the acoustic environment demands it. The concept of the functional unit of muscular contraction, the sarcomere, contains within it one of the most elegant solutions to a mechanical engineering problem: the need for a structure that can generate both the force required for maximal effort and the fine-grained control required for the most delicate movements, across a range of operating lengths and velocities, while remaining reversible, self-regenerating, and capable of adapting to changing demand over a lifetime. The sarcomere achieves the apparently contradictory requirements of maximum force and fine control through the simple but profound principle of massive redundancy and graded recruitment. Each sarcomere, individually, generates a maximum isometric force of approximately 1 piconewton per crossbridge, an almost unimaginably small force. But a single myofibril contains hundreds of sarcomeres in series, and a single muscle fiber contains hundreds of myofibrils in parallel, and a single muscle contains hundreds of thousands to millions of fibers. The forces of each sarcomere, summed across all these levels of organization, produce the force of the whole muscle, up to several thousand newtons in the largest muscles. The control comes from the ability to vary the number of crossbridges contributing to force at any instant, by varying the calcium concentration, by varying the firing rate and the number of active motor units, by varying the velocity of shortening. The force can be modulated from essentially zero to maximum with a resolution that allows the most delicate manipulation of objects and the most powerful athletic performance. The reversibility comes from the thermodynamic cycle of the crossbridge, which can release its load and reattach elsewhere, allowing the sarcomere to shorten and lengthen in both active and passive states. The self-regeneration comes from the satellite cells and the protein synthetic machinery of the myonuclei, constantly replacing damaged proteins and building new sarcomeres in response to the demands of use. And the adaptability comes from the responsiveness of the gene expression machinery to the mechanical, metabolic, and hormonal environment of the fiber. The sarcomere is not just a mechanical solution. It is a biological solution to mechanical requirements, and the distinction matters, because biological solutions can adapt, repair, and improve in ways that no purely mechanical system can. Stay a while longer. You have stayed with me a very long time tonight. Through the sarcomeres and the calcium switches and the motor units and the tendon springs and the cardiac muscle and the smooth muscle of every quiet internal function. 100 facts, and you have been here for almost all of them. I do not take that lightly. Time, given freely in the dark hours, is the most generous kind. If Relax with Science has earned a small place in your evenings, if these nights have given you something you would like to return to, subscribing is the simplest and most honest way to say so. I will be here. I always will be. And now, just a little further. We are almost somewhere soft. You have stayed with me a very long time tonight. Through the sarcomeres and the calcium switches and the motor units and the tendon springs and the cardiac muscle and the smooth muscle of every quiet internal function. 100 facts, and you have been here for almost all of them. I do not take that lightly. Time, given freely in the dark hours, is the most generous kind. If Relax with Science has earned a small place in your evenings, if these nights have given you something you would like to return to, subscribing is the simplest and most honest way to say so. I will be here. I always will be. And now, just a little further. We are almost somewhere soft. The muscle fibers mitochondria, the organelles responsible for oxidative phosphorylation and the majority of the muscle's ATP production during sustained activity, exist not as isolated individual organelles but as a dynamic, interconnected network that continuously fuses and divides in response to the energy demands and stress signals of the cell. The image of the mitochondrion as a static, kidney bean-shaped organelle is, in most cells, a historical simplification that modern imaging technology has replaced with something far more dynamic. In muscle fibers, the mitochondria form a reticulum, a three-dimensional network of interconnected tubular structures that runs throughout the fiber between the myofibrils, with branches connecting the intermyofibrillar mitochondria to a subsarcolemmal population lying just beneath the cell membrane. This network is in continuous flux. The processes of mitochondrial fusion, the merging of two mitochondrial membranes into a single larger mitochondrion, and fission, the division of one mitochondrion into two, operate simultaneously to reshape the network in response to the cell's energy status and stress signals. When energy demand is high and the mitochondria are functioning well, fusion predominates, creating a more connected network that facilitates the distribution of metabolites and membrane potential across the network. When the mitochondria are damaged or the cell is under stress, fission isolates the damaged portions, which are then removed by mitophagy, the selective autophagy of damaged mitochondria. This dynamic quality is important for the maintenance of mitochondrial function across the lifespan of the fiber. The continuous renewal of the mitochondrial population through the cycles of biogenesis, fusion, fission, and mitophagy prevents the accumulation of damaged mitochondria that would otherwise progressively impair the fiber's oxidative capacity. Endurance training increases both mitochondrial volume density, the amount of mitochondrial membrane within the fiber, and the rate of mitochondrial biogenesis, expanding the fiber's oxidative capacity through the creation of new mitochondrial material through a pathway activated by PGC1-alpha. The stretch reflex, the monosynaptic reflex arc that produces a brief muscle contraction in response to sudden muscle stretch, is the fastest neural reflex in the body, with a latency of approximately 20 to 30 milliseconds, because it requires only two synapses, the IA afferent synapsing directly on the alpha motor neuron in the ventral horn. The monosynaptic stretch reflex is the reflex arc that the doctor elicits with the tendon hammer, the patellar tendon tap that produces the knee jerk, the Achilles tendon tap that produces the ankle jerk, the biceps tendon tap that produces the biceps jerk. In each case, the sudden stretch of the muscle by the tendon tap activates the muscle spindle IA afferents, which carry the signal at high velocity, 70 to 120 meters per second, to the spinal cord, where they synapse directly on the alpha motor neurons of the same muscle. The monosynaptic connection, a single synapse between sensory and motor neurons, is what makes this reflex so fast. The signal from the spindle to the motor neuron takes less than a millisecond, and the total reflex latency from the tap to the muscle contraction is approximately 20 to 30 milliseconds, limited primarily by the conduction time in the afferent and efferent fibers. The clinical value of the stretch reflex is that its amplitude and character reflect the integrity of both the reflex arc and the descending motor control systems that modulate it. A hyperactive reflex, one that produces an exaggerated, brisk contraction or clonus, suggests that descending inhibitory control of the reflex has been disrupted, as occurs after upper motor neuron lesions. A diminished or absent reflex suggests either a peripheral nerve lesion interrupting the arc, or a lower motor neuron lesion. The stretch reflex is thus one of the most informative single physical signs in neurology. A small tap with a rubber hammer revealing the state of both the peripheral and central nervous system through the simple act of making a muscle contract. The muscle's ATP consumption during rest, which sustains the NAS/KAT passe, the circa pump, and the basic metabolic processes of the resting fiber, represents approximately 25% of the body's total resting metabolic rate, making skeletal muscle the largest single contributor to basal metabolic rate in proportion to its mass. The resting muscle is not metabolically quiet. Even in the absence of any contractile activity, the muscle fibers must maintain their resting membrane potential, a task requiring continuous NAS/KAT passe activity to counteract the constant leak of sodium into and potassium out of the cell through the resting conductance channels. They must maintain a very low resting cytoplasmic calcium concentration, achieved by the continuous, low-level activity of the circa pumps cycling the small amount of calcium that leaks from the sarcoplasmic reticulum back into it. They must maintain the protein synthetic and degradative machinery that turns over the contractile proteins continuously. All of these maintenance tasks consume ATP, and their combined demand means that even a completely resting muscle, one that is not contracting at all, consumes a meaningful amount of oxygen. In a 70-kilogram person whose skeletal muscle mass represents approximately 30 kilograms, this resting metabolic activity accounts for approximately 20 to 25% of the basal metabolic rate. This fraction has an important implication for body composition and weight regulation, because muscle mass is the largest contributor to resting metabolic rate of any tissue. Changes in muscle mass produce corresponding changes in basal metabolic rate. A kilogram of additional muscle mass increases the resting metabolic rate by approximately 13 to 15 kilocalories per day, a modest figure for a single kilogram. but meaningful in aggregate for individuals with large differences in muscle mass. The muscle, even at rest, is working. Even tonight, as every fiber is maximally relaxed and no cross bridge is cycling and no movement is occurring, the NAS/KAT passe is working, the circa pumps are working, the ribosomes are translating, the proteasomes are degrading. The resting muscle is not resting in any absolute sense. It is simply resting from the task of moving. The task of simply being continues. The three primary energy systems of muscle, the phosphagen system, the glycolytic system, and the oxidative system, do not operate sequentially but simultaneously, with their relative contributions shifting smoothly as exercise duration and intensity change, in a continuous, dynamically regulated metabolic response to the demands of the moment. The popular teaching that the phosphagen system powers the first 10 seconds, glycolysis the next 2 minutes, and oxidative metabolism everything beyond that is a useful simplification but an oversimplification. All three systems operate from the onset of exercise. The phosphagen system responds essentially instantaneously. Creatine kinase is already active, and as soon as ATP begins to be consumed, creatine phosphate begins to be hydrolyzed to regenerate it. Glycolysis begins within the first seconds of exercise, its rate increasing as ADP, inorganic phosphate, and AMP accumulate. Oxidative phosphorylation begins almost immediately as well. The delay of oxygen supply to the mitochondria is measured in seconds in well-trained muscle with a functioning cardiovascular system, not in the minutes that early models suggested. The relative contributions of the three systems shift continuously as exercise proceeds. At the very start of a maximal effort, phosphagen makes the largest contribution. As the phosphagen store depletes and glycolysis reaches its maximum rate, glycolysis becomes dominant. As exercise continues and the cardiovascular system delivers oxygen in increasing amounts, oxidative phosphorylation gradually assumes the majority of ATP supply. At any given exercise intensity, the steady-state metabolic response, the mix of substrates and pathways used, depends on the intensity relative to the muscle's aerobic capacity. Below about 60% of maximal aerobic capacity, fat oxidation provides a substantial fraction of the fuel. Above this, carbohydrate dominates, near maximum. Anaerobic glycolysis produces a significant fraction of the ATP. The muscle is metabolically flexible. It reads the signals of energy demand, substrate availability, and oxygen supply and continuously adjusts the flow of carbon through each pathway to match supply to demand as closely as possible. The loss of muscle mass with aging, sarcopenia, begins insidiously in the 4th decade of life and accelerates after 60, with the average person losing approximately 3 to 5% of muscle mass per decade after age 30 and 1 to 2% per year after 70, leading to the profound physical frailty that reduces independence and quality of life in older adults. Sarcopenia, from the Greek for poverty of flesh, is one of the most consequential changes of normal aging and one of the least discussed. It begins long before anyone notices it. Muscle strength typically peaks in the mid-30s and begins a slow, barely perceptible decline that accelerates in the 6th decade and becomes clinically significant in the 7th and 8th. The loss is not uniform. Fast twitch type 2 fibers are preferentially affected, being lost at twice the rate of slow twitch type 1 fibers in the early stages of sarcopenia. This preferential loss of fast fibers has direct functional consequences: the power output of muscle, which depends on the product of force and velocity, and which is disproportionately contributed by the fast fibers, falls faster than maximal isometric force, and it is the loss of power that most directly affects the movements of daily life. Rising from a chair, climbing stairs, catching oneself after a stumble, walking quickly, all of these require muscle power, the ability to generate force quickly, and all of them become progressively more difficult as the fast fibers are lost. The mechanisms of sarcopenia are multiple: reduced anabolic hormonal signaling, lower testosterone and growth hormone, impaired motor neuron function, denervation of fast-twitch fibers with incomplete reinnervation, reduced protein synthetic response to feeding and exercise, increased inflammatory cytokines, and reduced physical activity, creating a cycle of disuse atrophy. Resistance training is the most powerful intervention against sarcopenia. It recruits the fast fibers specifically, stimulates protein synthesis, and maintains motor neuron integrity. And it is effective even in the very elderly. The muscle of an 80-year-old responds to resistance training. It builds. The capacity for adaptation does not disappear with age. It simply requires a more deliberate cultivation. The smooth muscle of the blood vessel wall responds to the shear stress created by flowing blood by producing nitric oxide from the endothelium. The nitric oxide diffusing into the underlying smooth muscle, activating guanylyl cyclase, raising cyclic GMP, and causing relaxation and vasodilation, a mechanism that connects blood flow itself to the regulation of blood vessel diameter. Nitric oxide, once known as endothelium-derived relaxing factor before its chemical identity was established in the late 1980s by Robert Furchgott, Louis Ignarro, and Ferit Murad, work that won them the Nobel Prize in 1998, is one of the most important signaling molecules in the cardiovascular system. It is produced in the endothelial cells that line the inner surface of blood vessels by the enzyme endothelial nitric oxide synthase, ENOS, which converts the amino acid arginine to nitric oxide and citrulline. ENOS is activated by a rise in intracellular calcium in response to various receptor-mediated signals, and, crucially, by the mechanical stimulus of fluid shear stress acting on the endothelial cell membrane. When blood flow increases, as during exercise, when the cardiac output rises and blood rushes through the vessels at higher velocity, the shear stress on the endothelium increases, activating ENOS and increasing nitric oxide production. The nitric oxide diffuses freely through the endothelial cell membrane into the underlying smooth muscle, where it activates soluble guanylyl cyclase, increasing the production of cyclic GMP. Cyclic GNP activates protein kinase G, which phosphorylates multiple targets in the smooth muscle cell, ultimately reducing cytoplasmic calcium and decreasing myosin light chain kinase activity, causing relaxation and vasodilation. The vessel dilates in response to the increased flow. The mechanism is self-regulatory and continuous. The very fact of blood flowing through a vessel produces the signal that keeps the vessel appropriately dilated to accommodate the flow. The smooth muscle of the blood vessel, regulated by this flow-dependent mechanism, is always listening to the blood passing through it. The muscles of the tongue, the pharynx, and the larynx must coordinate with extraordinary precision during swallowing, a process that involves more than 30 muscles contracting and relaxing in a specific sequence over approximately 1 to 2 seconds, and that must simultaneously protect the airway from aspiration while propelling the bolus efficiently into the esophagus. Swallowing, deglutition, is one of the most complex neuromuscular programs in the body, and it is one that occurs largely below conscious awareness, several hundred times per day, during eating and drinking, and with each unconscious swallow that clears the mouth of accumulated saliva. The oral preparatory phase is the only stage under voluntary control. The jaw and tongue muscles masticate and move the food bolus into position. Once the voluntary swallow is initiated, the process becomes a reflex controlled by the swallowing center in the brainstem medulla. The pharyngeal phase, lasting approximately 1 second, involves the sequential contraction of the superior, middle, and inferior pharyngeal constrictors, which strip the bolus downward through the pharynx, the elevation of the soft palate to seal off the nasal cavity, the elevation and forward movement of the larynx, which opens the cricopharyngeal segment, the entrance to the esophagus, and simultaneously moves the entrance of the airway under the epiglottis, the adduction of the vocal cords to close the glottis, and the depression of the epiglottis to cover the closed glottis, providing a second layer of airway protection. The coordination of these movements, the airway protection occurring simultaneously with the esophageal opening, the bolus being directed precisely between the occluded airway and the open esophageal entrance, requires the precise timing of motor activity in the musculature of the pharynx and larynx that is the product of the swallowing center's pattern-generating circuitry. Neurological disorders affecting the brainstem or the cranial nerves innervating these muscles, stroke, motor neuron disease, Parkinson's disease, produce dysphagia, the difficulty swallowing that carries a high risk of aspiration pneumonia. The muscles that protect the airway with each swallow are among the most consequential in the body. The force produced by the heart muscle over a single lifetime, 70 years of continuous beating at an average of 70 beats per minute, amounts to approximately 3 billion contractions, each producing enough mechanical work to lift a kilogram weight approximately 1 centimeter, for a total mechanical work output of approximately 30 million joules, enough to lift a fully loaded truck. The arithmetic of the heart's mechanical output across a lifetime is one of those quantitative results that, when you work it out, produces a number that resists comprehension. 3 billion contractions. In each contraction, The left ventricle ejects 70 milliliters of blood against an aortic pressure of approximately 120 millimeters of mercury, doing mechanical work of approximately 1 joule. Over a lifetime, the cumulative mechanical work output of the left ventricle alone approaches 1 billion joules, enough to lift a 1,000-kilogram mass to a height of 100 kilometers. And this is only the left ventricle. The right ventricle, working at lower pressure against the pulmonary circulation, adds another fraction of this total. The energy comes from the oxidative metabolism of the cardiac muscle. The heart consumes approximately 8% of the body's total oxygen at rest, despite comprising less than 0.5% of the body's weight, reflecting its extraordinarily high mass-specific metabolic rate. The heart extracts approximately 65 to 75% of the oxygen from the coronary blood, the highest extraction fraction of any organ in the body, which means that the coronary circulation has essentially no reserve for increasing oxygen delivery by increased extraction. When the heart's oxygen demand increases during exercise, it can only be met by increasing coronary blood flow, which is why coronary artery disease, the narrowing of the coronary arteries that limits this flow increase, produces the chest pain of angina specifically on exertion. The cardiac muscle, the most productive machine in the human body, is also the most dependent on continuous and adequate blood supply. Tonight, it is doing its work at its slowest rate. Let it. It has earned the slow hours. The muscles of the body, taken together, represent approximately 40% of the body weight in an average adult male, roughly 28 kilograms of muscle in a 70-kilogram person. And this mass represents not merely structural tissue but a metabolic and endocrine organ of broad systemic significance, producing myokines that influence the function of virtually every organ in the body. The discovery that skeletal muscle produces and secretes biologically active peptides, the myokines, in response to contraction has transformed the understanding of muscle from a purely mechanical organ to a signaling organ with systemic effects. The first and most studied myokine is interleukin-6, IL-6, which is produced by the muscle in large amounts during exercise and whose plasma levels can rise more than 100-fold during prolonged endurance exercise. This exercise-derived IL-6 acts on the liver to stimulate glucose production, on adipose tissue to stimulate fat mobilization, and on the immune system in ways that, unlike the IL-6 produced in inflammatory contexts, are generally anti-inflammatory. Other myokines include BDNF, brain-derived neurotrophic factor, which is produced by contracting muscle and crosses the blood-brain barrier to promote neurogenesis in the hippocampus and support cognitive function, irisin, which promotes the browning of white adipose tissue and increases energy expenditure, myelin-like factor, which has anti-inflammatory effects, and many others whose functions are still being characterized. The myokine profile of an exercising muscle is, in effect, a molecular announcement to the rest of the body that the muscle is working, a signal that mobilizes metabolic substrates, prepares immune defenses, supports the brain, and stimulates the metabolic adaptations that make exercise beneficial for almost every organ system. The extensive health benefits of regular physical activity: reduced cardiovascular disease risk, reduced cancer risk, improved cognitive function, reduced depression, extended lifespan, are, in significant part, mediated by the myokines that contracting muscle secretes. The muscle, exercised, improves the entire body. Its secretions are the mechanism of its generosity. Every movement you have ever made, every step, every gesture, every breath, every heartbeat, every smile, every word spoken and every word held back in silence, was a muscular event, and the more than 600 muscles that have been serving you through every moment of your life are, right now, as you lie here, finally in the state they have been working toward all day, released, heavy, warm, and resting in the dark. Begin with the simplest version of this fact: you are made of motion. Not metaphorically, literally. The body that lies here tonight is a body built for and defined by movement. Every structure adapted for the generation, transmission, and control of mechanical force through the contraction of the muscular tissue that permeates it. The striations of the skeletal muscle that moved you here. The peristalsis of the smooth muscle that has been processing the food of the day. The continuous, tireless cycling of the cardiac muscle that has been circulating the blood since before you were born, and that will continue until the very end. All of this is muscle. All of this has been working. And now, with the body horizontal and the voluntary muscles finally released from the posture and movement and expression and speech that the day demanded of them, there is a particular quality of quiet that the muscular system settles into in the hours of rest that it approaches at no other time. The cross bridges have detached. The calcium has been resequestered. The motor neurons are firing at their minimum rate or not at all. The sarcomeres, those 2 micrometer molecular machines that have been the engine of everything you have done today, are sitting at their resting length in the dark interior of the fibers, surrounded by the mitochondria that have been powering them and the sarcoplasmic reticulum that holds the calcium they will need tomorrow. The muscles are not gone. They are not finished. They are simply, tonight, at rest. And there is a quality to this rest that is different from the rest of inanimate things, because these muscles will wake again. They will contract again. They will carry you forward into tomorrow and the day after and all the days that follow. Right now they rest in the knowledge, not conscious knowledge but the knowledge of biology, the wisdom written into the cycling of calcium and ATP in cross-bridge formation and release, that they will be needed again, and they will be ready. Let them rest. Let them be warm and heavy and still. They have done everything you asked of them today. And in the morning, they will do it again. The muscles you have been traveling through tonight are not separate from you. They are not a subject you have been studying at a distance. Every word of what you have heard tonight describes what is right now, at this very moment, in this bed, in this dark, doing the extraordinary, unremarkable work of being alive. The diaphragm that has been cycling all evening. The heart that has never stopped. The postural muscles that finally gave up their vigilance when you lay down. The smooth muscle of the blood vessels, still adjusting tone, still routing blood to the organs that need it through the sleeping hours. The muscles know this. In the cellular sense, in the biochemical sense, they are doing what they have always done, which is everything. Somewhere in the course of this long night together, the day has loosened its grip. The accumulated tension of the waking hours, the contracted jaw, the raised shoulders, the braced core that greets the demands of modern life, has softened. The muscles that hold the face and its expressions have gone quiet. The muscles of the hands, which have been gripping and holding and typing and carrying since the morning, have opened. The muscles of the back and neck and hips have surrendered to the surface that is holding them. This is not the absence of muscle. This is muscle doing something it does rarely in the waking hours, nothing at all. Simply being still. Simply being warm. Simply being the soft interior of the body that holds the blood and the heat and the breath, doing nothing except persisting, which is itself a form of extraordinary effort that you no longer need to be aware of. This is relax with science, saying goodnight, and meaning it with the full weight of everything we have moved through together tonight. The wonder that was here in the facts does not depart when the words stop. It remains in the muscles you just heard described, in the sarcomeres at their resting length, in the cardiac muscle cycling quietly in its chamber, in the smooth muscle of the blood vessels maintaining their tone against the pressure of the circulation. The miracle is not the facts. The miracle is the body they were about. The body you are living in. The body that has been working reliably through every moment of your life without ever asking to be noticed. You are the more than 600 muscles. You are the 3 billion heartbeats. You are the 20,000 daily breaths of the diaphragm. You are all of this, and you are resting now. Let the body be heavy. Let it be warm. Let it be exactly where it is.